This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 671, recorded on October 8th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York, Daniel Griffin.
Hello, everyone.
How's it going, Daniel? Short week. Only talked to you on Monday, huh?
We're talking a lot this week. I'm doing well. My wife told me not to say this, but I'm going to say it anyway. I'm feeling great. I got my flu shot. I feel 20 years younger.
Oh my gosh.
Something special in the flu shot this year. Everyone go out and get your flu shot.
Wow, I should do that. Yeah, I need to get it. I need to get the high dose though, because I'm over 65.
Yes, they did check my age, but not in the good way. Like, oh, you're so young. Are you allowed in the bar? It was like, are you old enough that I shouldn't be giving you the regular dose?
Yes.
So
I like your Parasites Without Borders bow tie, Daniel. Very nice.
Oh, thank you. Yeah, people who are watching can see I've got my, and I actually, when I'm in the hospital, I have matching socks, matching hat, and matching mask.
Nice. Very good.
Yeah.
Nothing like a coordinated doc to cheer up the patients, right?
Yeah, I think it does. The patients actually, patients are in the hospital and it's nice. I think it, they often comment, they often enjoy it. So,
all right, Daniel.
So
what's new this week?
Well, let's start with our quotation. Do not wait for leaders. Do it alone, person to person. And that is Mother Teresa. So People have probably heard of Mother Teresa.
Mm-hmm.
At one point, I was working in Nepal. I was working in Kathmandu at BR Hospital, and I was invited to go work with Mother Teresa. And what did I say? I'm much too busy. My life would have taken probably a slightly different turn if I had not been much too busy. Let's talk. I'm going to stay on my theme. I'm going to be positive, but informative. So I listen to the other TWiVs. Like, so when I'm not on, I was listening to them once they get dropped. And so I want to give a positive take on TWiV 669, those surgeons up from Boston. And they talked about how great it would have been if they had been able to hear from physicians in Wuhan instead of feeling like they needed to reinvent the wheel. And I was listening to that saying, well, guys, you should have, I guess, they weren't listening to TWiV back then.
Yeah.
But I actually was receiving communications from doctors in China. And I was looking through them after I heard that. It was great. I mean, there were discussions in there about neutrophil lymphocyte ratios, steroids, anticoagulation, proning, secondary infections, all this stuff, right? And this is back in March. So it was really, I have to say, it was really great. And I guess this is what made me think of the Mother Teresa comment. Mm-hmm. As physicians at a grassroots physician-to-phician level, these informal networks and sort of the informal network that you have helped us put together here, Vincent, I had physicians from throughout the world emailing me, emailing back, phone calls, texts. We were setting up the weekly Parasites Without Borders physician newsletters with hundreds of physicians, using this as a resource. And it was really a circular experience because I was hearing from physicians in China, in Italy, in France, in Africa, in India. And we were all sharing our experiences at the bedside and learning together. And a lot of what made this happen actually was TWiV, This Week in Virology, because everyone would know that everything we shared, I would get an opportunity to sort of put together and distill and disseminate, in our, initially our recordings from the parking lot with me sitting in a car, but then also put together in the Parasites Without Borders newsletters that would come out. So So perhaps many things went wrong, but there was still some stuff that went well. And my hope is that we've learned a lot from this. And I think this is one of the things that we've learned is, this is not a time to silo up. I mean, sometimes I would get like, these are, our guidelines here at our institution, but then the institution would be blacked out because they were not really supposed to be sharing for whatever legal fears. But we were sharing, we were communicating. So, and thanks to everyone, all the, Vincent and your team made this happen. All right. So as far as news, a bunch of stuff happened this week, right? A famous person got COVID. More importantly, ASH, the American Society of Hematology, announced its anticoagulation guidelines. So we're going to go there. But I'm going to talk about treatment and drug updates. And I think that what I've realized over this last week is, people really like a review. Where are we now? And I think that's what, we sort of got these questions coming up as they looked at what treatments are being done in a famous individual. How do these compare to the standard of care? And a lot of the questions I got, interesting enough, were about, trying to get together on a standard nomenclature for the phases of the disease. And if any of our listeners know Paul Merrick, I think he and I have decided to write a paper together with Stephen Dittmore and Ben No. And because some of us use slightly different terminology with regard to the phases. And I think part of it is based upon our background, our specialties. So I'm going to try to sort of put these together. I'm going to run through these as I talk about. So first there's before you get the disease. And I think that's pretty self-explanatory. You haven't been exposed, you're out there in the world. And what are the preventive strategies? So one of the things that a lot of people have been watching, right, are the vaccine trials. And we talked quite a bit about those. And as far as news, I think that there's reason for people to start to feel confident and positive. I think the development that with the announcement from the FDA really saying that, there's not going to be a vaccine released that is not going to be safe. And so, you hear all the polls, and the polls say, majority of Americans would not take a vaccine. But then in small print, they say, if rushed to market, if not proven to be safe, if endorsed by a politician and not by the scientists. But, in bold print, I think a lot of Americans, when they ask them the second question, they say, well, if it's available, do you want it? And they say, oh yes. So I think the nice thing we really got from the FDA, and the nice thing I think about having someone like Anthony Fauci of impeccable integrity is a couple months from now, we are, I think, going to have preliminary evidence. We're going to really start to get to a point where we're going to be looking at do these vaccines work, and really starting to see that efficacy and safety data starting to roll out. So I'll say as far as preventive therapies, this is getting closer and closer every day. Things are looking good. They've enrolled tens of thousands of people in these trials. So excellent stuff. Reason to be optimistic there for, and I'll name the front runners, right? Moderna, Pfizer, J&J.
Speaking of positivity, you listen to TWiV 670, Kizzmekia Corbett, who's been working on the mRNA vaccine at the NIH. She said it's an amazing platform. I haven't seen one example where it doesn't work, so could be right, Daniel.
I'm going to say don't listen, watch, because she's great.
Yes, she's very good.
No, actually, I enjoyed that episode, and I think that was part of it. Maybe I'm feeling, maybe it isn't the flu shot. Maybe it was enjoying that episode of TWiV that it just gave me this positivity. That or that dose of steroids I just took. No, that was a joke for all the nerdy scientists. The other things that there are some interesting studies, correlations, nothing compelling, but having a normal level of vitamin D and zinc, might be something worth doing. More and more data on something, and I'll pull it off the thing here. And for those of you not watching, I just put on my Doctor Who mask, which says masks are cool, the doctor. Masks work. They don't make you hypoxic. It is actually a very effective way to keep us safe, particularly as we're heading into these winter months. The distancing works, the hygiene works. Sorry to upset people, but you may need to keep washing your hands a little bit longer. Um, but then what about the viral phase? And I want people to think about this. So don't close your eyes and imagine this if you're driving, but if you're not driving, you can close your eyes and think about this. The viral phase. And there's two ways we talk about the viral phase. I look at Vincent because I think in his mind, when he thinks about viral phase, he's thinking about RNA copy numbers. They're really high and then they're coming down. At the same time as that is happening, there's the viral phase or the viral symptom phase that I think about as a clinician. As the virus is just starting to come down is when the symptoms are just starting to appear. So there's really two curves. There's what I call the playground slide, which is the viral levels going up and then coming down. But there's really a hill, which is the hill of viral symptomatology. So the virus is starting to go up a couple days before those symptoms appear. In general, it's going to last for a few more days. Most people have a infectious period of about 5 days, but that feeling crummy viral symptom phase, that's going to really last for that first week and kind of move into that second week when we'll start talking about the second phase. So what do we do during that viral phase? Everyone now is getting familiar with the monoclonal antibodies. And there's some promising data coming out of 3 companies, Eli Lilly, Amgen, Regeneron, but others are trying to get into the game. Just a clarification, Regeneron is a company, not a product. We did not give the president the entire company. We just gave him one of their therapeutic agents, which is two monoclonal antibodies. And if you look at the studies on this, and the studies, let's be honest, we're getting mostly press releases. We're not seeing, the level of peer review data that we really would like to see as these are being pushed for EUA. But these are targeting viral phase. These are antibodies targeting the spike protein. And some of the preliminary stuff is really saying you want to get this in that first week. And this will hopefully result in a reduction in people going to the hospital, a reduction in that antigenic stimuli that's going to lead to the later stages of disease. And we've been working a lot, our organization, with setting up some of these trials so people can actually get these. We can get answers to the efficacy behind these. And the safety, so far the safety, as we anticipated, has been really good safety profile, but small numbers, right? The president was, I think, an N of 10 who got it in compassionate use. There were only a little over 200 folks in the Regeneron data that we discussed that came out in press release form. So
now I understand that they applied for EUA, right? And that would be, I suppose they have given the FDA the real data, not just the press release, right?
You will need to give the real data to the FDA. And I don't know how many people in the FDA listen, but the FDA, these are bright scientists at the FDA. And actually they're starting to, I think, look at stuff in these phases and the idea of what works when. And I'm going to get to that as well. But yeah, they're going to want to see the data. And I think that's the FDA has really stood up and said, hey, we are an independent organization that will be making decisions and recommendations based on science, not politics. So I think everyone can feel really reassured there. Remdesivir, right? That's really our other therapeutic that's out there. It's really the one that's out there. It's the one that has some data showing that probably the earlier you give it, the better. We still wait in most situations until someone starts to require oxygen. And part of that I still think is a rationing. We're trying to make sure that we don't give out these, $3,000 a dose, medication courses to people who are going to get better on their own. But interest is also ramping up in the idea of maybe there's some way during this viral phase to ramp up the innate immune system. And there's a couple sides to this that I think either you've talked about it or you're going to talk about it. There was a couple interesting papers. One was where they looked at people with a type 1 interferon deficiency in the blood. And that was sort of type 1 interferon deficiency in the blood could be a hallmark of severe COVID-19. But then there was also another paper that Casanova's group, and I think he's still over at Rockefeller, and it was inborn errors, type 1 interferon immunity in patients with life-threatening COVID-19. And this paper, I think we're going to be getting up on the Parasites Without Borders website. But basically, if you saw people who had these rare variants that are predicted to give them a loss of function of interferon production, there tends to be an association in these individuals having life-threatening COVID-19. So some interesting sort of suggestion that maybe if you're not getting a good interferon innate immune response, that might allow you to progress, that you might not clear the virus as quickly. You might have all this virus and antigen that's going to stimulate the immune system. There was an encouraging press release that came out over the summer. This was this inhaled interferon, and just phase 2 at that point, small group, couple hundred patients. And when they inhaled the interferon beta therapy, there was a lower progression to severe disease. But a word of caution, and this is actually something that I'm sort of in an email chain here with the FDA about, is they just announced that they had stopped enrollment of severely ill COVID-19 participants in a study looking at subcutaneous interferon beta-1 alpha. And this is a drug that's approved for MS, multiple sclerosis. And the Data Safety Monitoring Board found when they looked at patients that were on high flow oxygen, non-invasive mechanical ventilation, so people late in disease, people who we would think sort of missed the window for ramping up the innate immune system, that those patients actually did worse. So they did not want that part of the study to continue. And that's actually part of this dialogue I'm having in the paper I'm going to write, hopefully with Paul Merrick and Steve Ditmore and Ben No, about, the timing of different drugs relative to the windows. You don't really want to ramp up the innate immune system during the first week, that viral phase. You don't want to ramp it up when you're starting to have the second week, what I call the cytokine storm phase. The pulmonologists call the pulmonary phase, right? If you got a hammer, everything's a nail. If you're a pulmonologist, everything's about the lungs. I say that with a smile, Paul Merrick. But yeah, so as an ICU doc, this is when you start seeing people having increased oxygen needs. This is when you start seeing the pulmonary. So as a clinician, you're seeing pulmonary. As an immunologist, I'm seeing the cytokine phase as sort of the underlying, but it's the same thing. It's phase 2. This is the point where now we have good evidence for steroids. And there was a nice publication. It was a meta-analysis in JAMA, association between administration of systemic corticosteroids and mortality among critically ill patients with COVID-19. And this looked at a number of studies, 7 studies, and basically reported a lower all-cause mortality with steroid use when you looked at all these. So at this point, we're still recommending routinely that individuals who start showing evidence of the cytokine storm, start moving into that pulmonary phase, that they get started on either dexamethasone, 6 milligrams POQ day for 10 days, or in some other studies, if they're sick enough to be in the ICU, sick enough to be on a ventilator, there may be some benefit to even moving up to higher doses. But the word of caution, again, this is not week 1. Week 1, if you give it to people while they're still trying to clear the virus, that may be harmful. Timing is really important. Tocilizumab, another publication, decreased mortality in COVID-19 patients treated with tocilizumab, a rapid systemic review and meta-analysis of observational studies. So, it's all in the title, right? This was a rapid review. It was a meta-analysis, observational studies, right? So we're missing the, randomized control, multi-center, placebo-controlled trial that I really want, that we really want. But they sort of calculated like what seems to be the benefit here based upon all these studies. And they were saying, if you figure the cost is about $500 a dose of this medicine. The number needed to treat to save one life was 11. So that's about $5,500 per life saved. There was another study, tocilizumab, for treatment of mechanically ventilated patients with COVID-19. And this had been published in CID. And that's the Journal of the ID Society of America, suggesting a 45% reduction in death when you use this with a background of steroids. So interesting. They did a subgroup analysis on folks that developed Staph aureus pneumonia. And it's interesting. The people who got the Staph aureus pneumonia, there was no difference whether they got TOSI or not. So even though this is something you might see, we can treat it. This reinforces what I've often said. I can treat a bacterial infection, but I can't treat death. So the IDSA has started to discuss, the ID Society of America has started to discuss TOSI. They're not routinely recommending it, and I'm going to say, nor am I. We sort of look at it as potentially having a role in patients who have gotten steroids, look like they're progressing, something to be considered. But boy, we're waiting for all the really good data to help clarify this. But now, what we're all excited for, anticoagulation. So just before we are recording this, Vincent, I was on the Zoom presentation of the American Society of Hematology, presenting and releasing for public consumption their guidelines. And what basically, and I'm going to just sort of give them right here. So the ASH guideline panel, and it's crazy, which reviewed like 50,000 publications and whittled it down to like 50. And basically, so guidelines suggests using prophylactic intensity over intermediate intensity or therapeutic intensity anticoagulation in patients with COVID-19 related acute illness who do not have suspected or confirmed VTE. And then we put a remark, and this remark is important. The panel acknowledges that higher intensity anticoagulation may be preferred in patients judged to be at high thrombotic risk and low bleeding risk. So let me translate that for the non-clinicians. So this guideline is now up there, basically saying and this reinforces what ASH and the American Thoracic Society has said for a while, but now it's formalized. Put everyone on anticoagulation, and folks that you judge to be at increased risk for thromboembolic complications and at an acceptable bleeding risk to do that, because there is a risk. These folks, you might consider going up to full dose. And so I'm going to throw, as you'll see, my patient updates at the end here now that we have this context. And let's do that. So now we're going to slightly out of order, but I do this on purpose. I wanted to discuss one of our patients that people have been following. And this is the woman who I presented, who had gone to visit her daughter in Virginia. When she got back, found out her daughter had COVID. She then herself got COVID, came in early. We gave her remdesivir. We gave her steroids. She was getting better, developed pneumonia, started her on antibiotics. We were thinking everything was great. And then all of a sudden, her hematocrit, her amount of red blood cells, dropped in half. Her left thigh got huge and swollen. And I want our clinicians to think about what complication you might have in a woman who is on the recommended anticoagulation to prevent the thromboembolic complications. So she did. She bled half of her blood volume into her left thigh, into her left leg. And so, hopefully everyone raised their eyebrows there. And this is sort of the whole, everything we do has a potential harm. So you've got to be balancing that. We transfused this woman. We discontinued her anticoagulation completely. Today, when I went and saw her, actually, I should say yesterday when I went to see her, she was clear. She was back to baseline. The bleeding has stopped. And I think she's going to do well. So this is why I think these guidelines are important. You need to be realizing that everything we do has potential benefit, but it also has potential risks, as we saw in this context. Another case I'm going to point out, and then we'll get into our tale of COVID. This is a young woman. I guess I shouldn't use the word young. I used young once on TWiV, and they said, that's not young. But this is a 32-year-old individual who works at a group home in Queens. And she takes care of a particular client who has a lot of needs. So it's a one-on-one relationship here. And in the end of August, her client was diagnosed with COVID. A week or so later, she started to feel not well. this progressed, but she stayed home. She got through it. Initially, she said the first week she felt just kind of crummy, fever, body aches. Second week started to have some trouble breathing. And then it was really the end of the third week when she comes to see me, when she's starting to have pain and issues with taking deep breaths. So we do a COVID-19 PCR, which is negative, but her serology test is positive. So this is that challenge, right? Very, very good, very compelling story, but really important to think about what's the right test for what I'm trying to see. So here's someone who's probably been sick for over 3 weeks, probably at the point where there's not going to be a lot of virus around, but probably at the point where we're going to start to see the antibodies come up. So she is doing well. We actually just discharged her today. So, all right. Understanding the tale of COVID-19. There was an article published in Clinical Microbiology and Infection, follow-up of adults with non-critical COVID-19 2 months after symptoms onset. And here they noted that more than a third were still sick. And so I think this reinforces the tale of COVID-19 is not an exception, but actually a very common part of the clinical course. And I just did a talk on Wednesday morning for all the pro-health doctors. We sort of have this Wednesday morning check-in where we kind of give updates on things. And one of the updates I did was talking about the tale of COVID and what we as clinicians can do. So I guess the pro-health docs, you're listening twice. You get to hear me twice, actually 3 times in 1 week if you listen to the presidential one as well. But what I talked about on that call was that thousands of people, thousands just in the New York area alone, had COVID and are now suffering with prolonged fevers, fatigue, insomnia, chronic cough, these thick pulmonary secretions, hair loss, brain fog, joint muscle pain, these hot flushing issues. And we can actually help these people. And the first thing as clinicians that we've been doing, and it makes a difference, just validation, just saying, hey, this is, you're not making this up. We're seeing this, going through some of the symptoms, even sort of letting them know, like, have you had this? Have you had that? And just the eyes light up with, oh my gosh, yes. How do you know? Second thing, we can actually do targeted therapy. We're having success using non-steroidal anti-inflammatories, things like Aleve, things like ibuprofen for the muscle and joint pain. Melatonin and antihistamines for this insomnia. Insomnia is a big part of this. Scheduled Tylenol for these prolonged fevers. A lot of these people, it may just be a low-grade fever, but every time they have that fever, it's exhausting. It's a reminder. Mucomist, I give credit to my pulmonary colleague, the N-acetylcysteine, helps with these thick pulmonary secretions. Gradually increasing the exercise for this post-exertional malaise that they get. Sometimes working with physical therapists to help with that as well. Frequent visits can help, as well as in a lot of cases, mental health referrals for the depression, the anxiety, the palpitations, all the other things that come with this. So I always want to leave, a word about the people with COVID so that people don't just forget about those folks. So that is it for me, unless do you have any emails for me?
I do. I have one from John, who's a hospital physician in New Mexico. He has a policy question. He's drunk the Kool-Aid about rapid tests. Closest test I've seen so far is the Binax Now, $5, not cheap enough, but close. The other problem is that the EUA from the FDA for this test and the information from Abbott states it's to be used for symptomatic people as opposed for screening. Why is that? Dr. Griffin has mentioned large screening initiatives by ProHealth in the New York area. What tests are they using? The EUA for ID Now is also restricted to symptomatic patients. These restrictions don't appear to be science-based. Is there any way around them?
This is great that this email is here because it sort of gives us a chance to talk about testing and what we do with that. I got a chance to enjoy, and I got a chance to inflict this on my family. But we have up and running our calculator, right? You can plug in our test calculator and say, well, which test might I consider doing? What's the cost of that test? What's the sensitivity of the test? What's the background prevalence? And you get to go through and play around with, what if I use let's say a PCR or a TMA, one of these very sensitive tests, and then you get to put in resulting delays. Am I going to find out in 1 day? Am I going to find out in 2 days? And it really, this is great because it gets you play around and you're like, oh my gosh, I guess that's true. And so, I started off with okay, we're going to test once a week. We're going to use the expensive PCR. This is going to be our background prevalence. How many infections are we going to see? And I used William and Mary as my, as my school, because I had my daughter Daisy saying, and I asked her, how many kids right now at William and Mary, College of William and Mary, are positive? Plug that in. How many people are in the school? Plug that in. And you do it, you say, okay, if we do the PCR, and then we have to do confirmatory testing for the false positives, those indeterminates, we're gonna see this many infections, and at a cost of this for every infection prevented. And then you go down, you say, well, what if I do these inexpensive tests? What if I do the $5 test, but I can do it every day? And it's amazing. You start getting down to levels where it's about $50 per infection prevented versus $5,000 per. So it becomes so much cheaper to use a less sensitive test, but more frequently. So this paper's out actually at New England Journal. The editor sent it out to the reviewers. So if any of the reviewers are listening, come on, get it back. We'd love to make this this calculator, open access so that people can jump on and play around. But I think that's part of the way that you start to really get it. You take a real concrete scenario. This is the scenario I'm looking at. You plug in the different options, and then you just start seeing like, wow, that makes sense. Now, the EUA is like so many things in medicine. It's not a restriction. No one's handcuffing you. Once it's out there, once it's available, a physician, a healthcare provider can actually make a decision. And so if someone's been exposed, if it's screening, which is what that would be, you can use an Abbott ID now. No one's going to come along and say that, ooh, that doesn't strictly follow. So there's no restriction. The EUA is not tying your hands. It's guidance, really. It's getting it out there. So what are we doing in the New York area? Just for specifics, this was on the Wednesday call. So all the pro-health physicians already know this. We have 50 urgent cares now in just the New York area. I guess we have more if you count the tri-state. Every one of the New York pro-health urgent cares has multiple Abbott ID Now machines. We also have the BD Veritor system rolled out at all the clinics. And this is that it's $20 a test, and there's a little machine, almost like a urinalysis we use. We're looking at moving forward with the Binex now when we get access to that. But as people probably are aware, most of those were bought up by the government or being distributed that way. And I think just the technology is going to move forward. For a lot of our large screening programs, we still are using PCR, and it's kind of funny. Some people can say it's almost like flying first class. When you offer them other options, I want I hear the PCR is what I want. And it doesn't matter. You can charge them $100, $200. They'll pay out of pocket because it's first class. So I think that we have to be comfortable using lots of modalities. You've got to really, and this calculator is great for that, you've got to say, what frequency of testing can I potentially do in a certain place? Right now, we're working, actually a contact from Mount Sinai, and this is a school outside of the city in a really incredibly poor area where it's part of our Let's Give Back program. In some schools, you can do it every day. Sometimes we break it down and you're not doing everyone every day, so it becomes every other day. But I think at the end of the day, we're using all the modalities we have. We're using PCR. We're using the template mediated. We're using antigen testing. We're using Abbott ID Now. And when we don't have enough capacity in the New York testing, which we don't always do, we actually have a fleet of planes. This is crazy at Teterboro. And every 2 or 3 hours, a loaded plane is flying to North Carolina where there's a fleet of machines down there doing more testing. So the amount of testing we're doing in New York is really, really incredible, right? It's over 100,000 a day. And I think that this is helping, but it's not helping as much. People need to wear their masks. They need to help us as well.
All right, so we had a lot of questions about the presidential episode. I picked just a couple here. First one is from David. The question is for Daniel Griffin. Is it unusual that Trump hasn't been given an anticoagulant other than aspirin? What's the reasoning behind this?
Yeah, this is an excellent question, and and it's very timely, right? We just came out with the American Society of Hematology guidelines. I don't even know if I was supposed to be telling anyone I was on that panel. I think I was sworn to secrecy, and we know how good I am at that. But when you look at all the data on anticoagulation, it's tough. It's low quality. It's not the kind of data you want. So it's very clear if you do nothing in a person who has COVID-19. And, the term severe is anyone who has hypoxemia, whose oxygen level drops into the 80s, not necessarily the low 80s, just any 80s, mid or high 80s, which is what I think we've understood about the president. That person is at an increased risk. And it's a really broad range. Maybe 20% of these people will have clotting complications if you do nothing. And so now the next question is, what is that something you do? And I think we related, if you use full dose anticoagulation, if you use prophylactic anticoagulants, these are the injectable low molecular weight heparin, some of the oral pills, we call them DOACs, which would be Eliquis and Xarelto. Some of our listeners have heard those. Or in someone who, and I think in this case, it's reasonable to say, here's an individual who is going to be ambulatory, he's going to be moving around, and so that may have been the decision behind the aspirin. I mean, his hospital stay was very short. He was not laying there in bed immobile. So, the walking around, the up and down the stairs, this may have been a risk-benefit decision that was made by his physician.
Steve writes, you were trying to explain early Decadron in Trump. Did you consider he had a transfusion reaction to Regeneron infusion, which caused pressure drop, a little pulmonary wheeze, mild desaturation? That might be why he was transferred to Walter Reed and given famotidine and steroids and not in the second-week protocol. Interestingly, Decadron was not mentioned as ongoing therapy with 5 more days of remdesivir. Just a thought. Might look bad to have reaction to compassionate use drug in stage 2 testing for doctors or Regeneron and skew the trial results.
Yeah, no. So Steve, these are good thoughts, but I'm going to clarify. So the agent, the Regeneron monoclonal antibodies that the president was given, these are 2 monoclonal antibodies. So it's not plasma. There's no serum. There's none of those factors that would cause a transfusion reaction. So what I think a lot of us have looked at this and said, we've now seen millions of cases, right, of COVID-19 in the U.S. And some of us, like myself, have treated hundreds of cases, over 1,000 actually myself. And there is a certain progression that we see. And this is actually the first week is when a person has viral symptomatology, when the virus is on the way down. It's really about day 7, day 8. It's really as you enter that second week that you start to develop low blood oxygen, so the hypoxemia. And the hypoxemia is the first thing you see, followed by increased respiratory rate. And so when the president dropped into the 80s on Friday, which is what we've now been told, I perceived that had to be day 7 or 8. So he was entering the second week. So I don't really think that the Decatron was given early. And I think I mentioned a little, he may not have noticed that he was feeling under the weather. This is a guy who, travels through different time zones. He's working a lot. He may have thought, I feel kind of tired and run down, but he may have thought, that wasn't because I'm sick. So I think my suspicion, a lot of our suspicion is that, he probably had been suffering from the virus, that Friday was actually day 7 or 8, and that the dexamethasone was given just at the beginning of the cytokine storm or the pulmonary phase. And that I don't really have any concern that was a reaction to the Regeneron product.
All right. The last one is from Robert, sort of along the same lines, who says, I understood from Dr. Griffin that because the president received Dex on October 4th, this suggests he was already in roughly the second week. Working backwards, this suggests he was already infected around the time of the first debate. Can we assume he's tested regularly and knew he was infected during the debate? Did the public only learn about the infection when it became clear he was symptomatic? These are rhetorical questions that I fully recognize don't require your expert knowledge of virology to answer. The only other alternative I can see is that he received Dex earlier, but Dr. Griffin says this is a no-no. So I can't imagine the president's medical team would have done this.
Yeah, that's entertaining. Yes, and I think we talked a little bit about his medical team last time, and so the physician, sort of the presidential physician, is a DO. And I talked a little bit about there's two tracks of training. There's the MD training, which is a type of the allopathic medicine, which is sort of I'll call it a less holistic, and the DO sort of came from more of a hands-on, and I say a lot of the DOs that I know view it as sort of a more of a holistic, broader training. And so his physician is a physician. His physician had the backup of really some excellent ID and critical care doctors. I don't think they're going to give this man steroids at the wrong time. I mean, I would find it a lot harder to believe that these really competent physicians would have done that. So yeah, it makes a lot of sense to me that this was day 7 or 8 when the oxygen dropped, and actually was appropriate timing for the steroids. And I should tell, steroids are interesting. I don't know if our listeners have ever gotten steroids, but it does raise some interesting issues. I mean, the president came out and said, I got the Regeneron and I feel great. I feel like I'm 20 years younger. I'm full of all this energy. And I was kind of a little worried because one of the known side effects of steroids is this euphoria, this sort of hypomania where you just feel great. And so, and I was talking to a buddy of mine, Adam Fitterstein. He's actually the head of the urgent cares here in New York. And he was taking care of it was an ICU nurse who, needed steroids or recommended to be on steroids for a certain indication. And she felt so good. She had this just wonderful, I'm going to be able to do anything I want. So she resigned her job. She put her house up for sale, and she was in negotiation to buy a van to become an itinerant dog groomer. And then fortunately, she finished her course of steroids the day before the open house and was able to cancel the open house and apologize to her coworkers, get her job back. But, the combination of, the COVID-19 can have some cognitive impacts. The steroids can give you this feeling like you're 20 years younger. I mean, so that was sort of my take on his comments, because, getting COVID-19 does not make you feel 20 years younger, but some steroids might.
Is it possible he just had a rapid course? I mean, I find it hard to believe that he would be positive and debate Biden. I would think his staff wouldn't permit that. Could he have been infected afterwards and just developed more serious disease more rapidly?
I think as we've talked about, the first week is really a viral symptomology phase,
Yeah.
And a lot of us, if we feel like we're fighting off a virus or we just feel like we're a little tired, we might continue our daily routine. It does raise the question of, does he get, frequent high quality testing? And suggestion is not. I know he arrived a little bit late, so he couldn't get the testing that was being offered at the debate. So not sure exactly when the last confirmed negative test was. But
all right. Okay, that is our weekly COVID-19 clinical report from Dr. Daniel Griffin. Thank you, Daniel.
All right. Thank you, Vincent. And everyone take care and wear your masks. Remember, masks are cool.
Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's 58 Fahrenheit, clear blue skies, mild winds, which is very nice because we had a squall blow through that was about 5 minutes long on Wednesday, and it knocked power out for 2 days. But none of that today. It's gorgeous now.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 80 degrees, overcast, headed for 85. The longer range forecast says it's going up to 96 again on Sunday, but then it's cooling off again. So we're not quite out of the woods yet. And my grandchildren tell me that outside currently there's about an 80% chance of mosquitoes. So they chased them. They went out to play for a little while in virtual recess for a recess from virtual school, and got chased inside by these insects.
Our guest today is back for a 4th TWiV. She is an assistant professor here at Columbia University. Amy Rosenfeld, welcome back.
Thank you. Thank you for having me. I'm in the annex, so I don't get to report the weather. I'm just in the annex of Microbe TV.
But if you yell loud enough, we can hear you in Vincent's office, right?
Probably.
Right. It shares a wall.
Well, the original plan was to have her here, but it was too much echo. But if you look behind me, there are pictures on the wall. Amy is right behind that wall, right?
Yes, I'm in the annex.
In the former, no, I don't own that. TV. We don't own that. That's the former office of Saul Silverstein, who was on TWiV number 8, I think.
TWiV. Yeah.
Number 8. And I have to say something.
Thanks Saul for contributing to the annex.
Yes.
Okay.
Yeah.
Welcome back, Amy.
Thank you.
Now, Amy has a lab technician position available to work with her on plus-strand RNA viruses that lead to respiratory and neurological disease. Applicants should have a BS, Bachelor of Science, not the other BS, that Bachelor of Science, some lab experience that is not a laboratory class. If you're interested.
Yeah, that's actually really important because lab classes, their experiments are always going to work. Whereas here,
Yes.
no,
They're
it's
hardly
true.
ever going to work in a real lab.
Exactly. And that's a big thing for people to get over and reconcile with so that they have to know how that.
I never got over it.
No.
Yeah.
You know, it's funny. Freshman chemistry in college, we were doing some distillation, this multi-step thing. Maybe it was a Grignard synthesis. I don't know. It was fun. And the girl next to me, you know, you distilled into a little glass thing and you had to clamp it because as it filled, it got heavier and she didn't clamp hers. And about halfway through, psh, and I didn't tell her. I felt really bad. I didn't warn her, but I didn't want to, you know, interfere. It broke. And
Well, that's better than my 8th grade teacher. We decomposed mercuric oxide
Yeah.
in the 8th grade where you boil off the oxygen. Yes. I took advanced AP chemistry and the tube was like this. You had the Bunsen burner. Guy came to look at the tube. He dropped it on my wrist.
Your
I have
wrist?
a lovely third. Yeah. I
Is
have a
your wrist
lovely
okay?
3rd degree burn.
Wow.
Sorry.
Anyway,
Anyway,
I did a lot of advanced chemistry experiments when I was a kid, but they weren't generally in classrooms. They were just to
show that
up in the basement, and
Okay.
just to show that
you know, coming from
no. I was not making anything to sell. No,
Right.
No.
Coming from the 60s, I had acquaintances who did laboratory home chemistry for a different purpose.
I did know a guy in college who paid his way through school with some off the books chemistry work.
talking
but
about
that's
Breaking
a different
Bad or
subject.
something like that.
Yeah, a little bit like that. It was not quite the same substance, but
Anyway, just
that
to
would be
point out
good.
that not all laboratory class experiments work, but they should.
right.
But it's because you made the mistake. Whereas
Exactly.
here, I don't know why
Frequently, it's
why
just
it doesn't
doomed.
work. If
Yes.
my
Yep.
hypothesis is completely wrong and we need to step back and reassess, or you forgot to add Taq polymerase.
What did you say, Alan? Doom?
You're doomed. Some experiments, you're doomed from the start. It was never going to work. So you didn't know that. going
Anyway,
in.
if you are interested in this position, you send an email to tech, TECH at microbe.tv, and that will go to Amy and she will get back to you, right? Right, Amy?
Yes.
You'll get back.
Yes.
And in addition, the Biological Safety Program at Columbia has an opening for a biological safety officer in the age of COVID-19. Biological safety professionals have never been more needed, valued, or felt more purpose in their calling. The ideal candidate would be someone with a life sciences degree, bachelor's or higher, and at least 2 years of laboratory experience who is hungry to learn about infectious agents and molecular biology. More information and application instructions are available on the Careers at Columbia website. Once you're on the site, search the staff job listings for biosafety, or we will provide a link in the show notes. And finally,
Or they can just send their application to me and I forward them to Chris.
And what you could actually do, since the qualifications for these are kind of similar, is apply to both jobs, right?
Well, that's what they do.
Yeah.
they could do that.
That's what I've had some who, before we worked out our financial difficulties, I forwarded to Chris.
And the last announcement, throughout the months of October and November, all donations made to Parasites Without Borders will be matched and doubled by Parasites Without Borders and donated to Microbe TV. Parasites Without Borders is the 501(c) nonprofit established by Daniel Griffin, and we are very grateful for his support. And you may ask, listeners, why we're giving you money? Well, I appreciate greatly all the money our listeners have given, but only about 1,000 of you are actually donating, and that's not enough to do what I want to do. So we search for alternate opportunities. Now, Parasites Without Borders is a 501(c). That means your donation to them is tax deductible. And then hopefully by the time they give it to Microbe TV, we will also be a 501(c). I've been working on that for a couple of years now. I'm not actually so good at the business part. But it's going forward. We are incorporated in New York State now as a tax-free entity or whatever it is. And now the IRS is next. And so that's my second foray into raising money, hopefully for a charity. Then other charities who can only give to charities will donate. And then if that doesn't work, we'll have to do ads.
Really? I can't just sell you to the highest bidder?
It doesn't work. But you may ask, what is it that I want to do? I want to have a studio and hire people to help and do all the stuff in a studio as opposed to here and at home, and have people who are invested and help out and bring this forward. So that's the goal.
Yeah, you want to do the stuff that the big professional podcasts do. Like if people listen to, Leo Laporte has a whole empire of these. So that kind of thing. He's got people.
People.
Yeah.
Now he and many other podcasts use advertising to fulfill. We used to do ads. We stopped, but I'm hoping not to have to do that. But if we have to do it to achieve our goals, we will. Anyway, we do thank all of you who have donated, and if you would like a tax-deductible donation, you can give to Parasites Without Borders. Thank you, Daniel, for doing that.
Yeah.
As we announced on Wednesday, in fact, the prize was announced on Wednesday just before we started recording. Nobel Prize in Chemistry to Jennifer Doudna and Emmanuel Charpentier. Now, on the website of the Nobel Foundation, it says, for the development of a method for genome editing. And, of course, that is the use of CRISPR-Cas systems to edit genes. So let's talk a little bit about what is CRISPR-Cas9, bacteria and archaea.
Because it has a lot to do with viruses.
Yeah, this is an ancient system of defense in bacteria and archaea. And new listeners may not know what archaea are. They are not bacteria. They were once thought to be bacteria until Carl Woese did some ribosomal RNA sequencing, and he said, these aren't even bacteria in a classic. If he had lived.
They aren't like any other form of life.
If he had lived, he would get a Nobel Prize, I think, because now the three domains of life, eukaryotes, bacteria, archaea, it's up to him, right?
Yeah.
Anyway,
yeah, he completely reconfigured the tree of life, and that was a really big deal, and he probably should have gotten a Nobel Prize for it. But
he died.
They don't do them posthumously, and he's gone now.
Anyway, the CRISPR-Cas systems, they're more than one. They are ancient defense systems in which, when you're infected with a phage or some foreign DNA comes in, pieces of the genome are stored in arrays in the host genome. If the host lives, of course. If you die, it's never stored. But some fraction live after invasion. And then they're transcribed into short guide RNAs, single-stranded guide RNAs, which will then attack incoming virus or DNA, which may happen later. And together with nucleases and other RNAs, they will destroy it.
This is kind of mind-blowing because we never really thought of bacteria as having immune systems, and then people discovered restriction enzymes, which are these, they recognize, this 6 or 8 base or 4, whatever sequence and cut it. And that's kind of it's an innate immune system that particular 6 or 8 bases doesn't occur very often in the host genome, but it may occur in the incoming genomes. And so that was what we thought of as bacterial immunity until this CRISPR-Cas system got worked out, which it turns out there's an adaptive immune system. Bacteria can remember past infections. I mean, I'm anthropomorphizing, but effectively what's going on here is the past infection causes this record of it to be inserted into the genome, a little sequence, and it's not 6 or 8 bases. It's a long enough piece that only that piece of DNA will ever be recognized as that thing. Just like when you get a vaccine against measles, it doesn't immunize you against polio. You're immune to that one thing. So it's an adaptive immune system in bacteria and archaea, which means that this goes way, way back. This has to be the dawn of immunity.
I want to editorialize archaea here just for a second.
Sure.
And that is that they are unicellular organisms without a nucleus, which is why initially they were confused. They were thought to be bacteria. Mm-hmm. And it wasn't until with Wose's work that you got down to the molecular level that you realized that in terms of evolution, genetics, molecular biology, they were a whole different thing. Okay. But to the untrained eye, they would look like bacteria.
Now, a lot of people worked on this system before Doudna and Charpentier. In fact, 93-ish, it was the first discoveries, and people found that this was an immune system in bacteria could protect against invading phages and DNA. And along the way, people began to understand that there are enzymes associated with these. There are Cas, CRISPR-associated enzymes. And I should tell you, of course, that CRISPR stands for clustered, regularly interspersed short palindromic repeats. And that's a reflection of these repeat sequences that are in the genome that are parts of the incoming viruses and DNA. And even Eugene Koonin was part of this. He said, where these things are, let's look next to it, because usually genes that have similar functions in the same pathway are near each other. And indeed, he saw next to them were the Cas enzymes, the CRISPR-associated nucleases, and those chop up. And people found that, okay, you have these records of infection, they're transcribed into RNA, and then there's another RNA that helps bring the nuclease to the target DNA, and that cuts it. And you introduce a break, and that inactivates, of course, the phage or the incoming DNA. And then people started to realize, we could use this for gene editing, because if let's say you make an RNA for a gene you want to disrupt, it'll get cut, and then it will be repaired by the cell, and that'll introduce an insertion or a deletion of some sort. And then people said, if we include, let's say we want to replace a gene, we could include in this with the enzyme, the Cas9, the guide RNA, the other RNA called the tracer RNA. If we add a gene that we want to put in, homologous recombination will add that gene back in. So people started thinking about gene editing. And what Doudna and Charpentier did, which was really cool, they said, let's combine the two RNAs into one. So we'll take the RNA that you use to target your gene and the other that the Cas9 needs and put them together in one. And it worked. And that is their contribution. And I think that's an important contribution. I just want to point out, though, that if you go to the Nobel site, they claim that they discovered CRISPR-Cas genetic scissors, they say, right? They say Charpentier and Doudna discovered the scissors in 2012, and that's not correct. They did do this combination of the tracer and the guide RNA. That's fine, and that's a revolutionary modification that makes it really easy to do this and do gene editing. But I'm sad a little because the Nobel has a wide reach, and people are going to think that they discovered it. And all these people that went before them, they're going to be lost in the wind. So that's my
one
only
of
objection.
the parts of this story that I really love is that along the way in the history, I used to think it was the initial discovery, but no, it's on the pathway of discovery. Is some work from scientists from Dan and Yogurt.
Yes.
Yes.
Okay. Which I just love this story because, to make yogurt, you need bacteria, and their bacteria are getting trashed by phage, and they want to know how they can make their bacteria resistant to phage.
Yep.
So they want to know bacterial immunity, and they studied this and made some seminal discoveries about the CRISPR system. There's this big science paper that's authored by all these people from Dan and Yogurt.
It's
terrific.
Yogurt. And what I think is so great about that is there's this common perception that basic research is those eggheads in the lab doing weird stuff that doesn't have any application. And
that's you, Amy.
Industry is where, yes. Industry is where people take that stuff and turn it into something useful, right? And here with CRISPR-Cas, you have this beautiful story where people in industry, Dan and Yogurt, I mean, they're just trying to put stuff in cups to sell it. They did the basic research. They found this thing and they said, hey, this is kind of cool. We can't really use it, but, we'll go ahead and publish it. And then the basic researchers in the academic lab came along and said, you know what you could do with that? And did the practical work and turned it into something that could be used for gene editing. And we should also probably mention Zhang along the way here too, who's, if Vincent is a little annoyed that the Nobel Committee phrased things badly here, I imagine how
he must be upset.
Zhang feels. Who there's a vigorous patent dispute going on now between the two groups that claim to have reduced this to practice.
I'm sure he and George Church are upset because I think they collaborated, right?
Yes.
But for years, people have been saying, well, so many people have been involved in CRISPR discovery. Who's going to get the prize? And this really makes it simple because the prize is really for their development, which made it facile to do gene editing. Right. It's not for the whole discovery. It's just for that, which is the most important. And now everyone's using that.
The thing with the Nobel Prize is this whole notion that we can pick 3 people who did the thing. That happens so rarely in science. It's always you get 4 or 5 different groups independently doing the same thing that leads to the breakthrough because the technology gets there, the ideas get there, and something gets discovered multiple times. And whoever happened to be a week earlier gets the credit and the Nobel. So there is always this arbitrariness, sometimes more so than others, to the Nobel Prize.
The other thing I want to point out is that if you drill down in the Nobel Prize, nobelprize.org site, you can always find a more extensive document, more extensive than the press release, that usually quite clearly and thoroughly describes the development of the research and gives references to all the people involved and at least implicitly, if not explicitly, rationalizes the choices that they made relative to other people. So maybe we could put that link in the show notes. It's a PDF.
Yeah.
Although they don't always say why they did not give it to people. And that's why Erling Norby goes into the archives, which open up 50 years after the prize. And one of the first things he did was to say, why didn't Jonas Salk get a Nobel Prize for the polio vaccine? And you can read the deliberations. They say, we don't think he did anything important.
Yeah, you have to read between the lines in these documents and come up with your own conclusion as to why the decisions were made.
Yeah.
I think this is all very hard because we all stand on the shoulders of others, right? And you can take
even Isaac Newton, who was not a modest man, said that. Yes.
Yes. Oh, really? He wasn't modest? No.
No. Oh, no.
You can take.
That might not have fit in the whole room.
Okay. Even you could pick any prize. So the one this week for medicine, the Hep C Discovery.
Yeah.
A lot of other people worked on other hepatitis viruses before that to set the stage. Even reverse transcriptase, which went to Temin in Baltimore. They were basing it on many years of work on tumor viruses, which led to this idea that they're permanently altering the cell. Something's got to be going on, right? But that's what a Nobel is. You may not like the idea of narrowing it to a few people, but that's what it is.
This is what Alfred Nobel wanted to do because he subscribed to the great man idea of history, and he wanted to try to renovate his legacy, having invented one of the most useful weapons of war in history.
Dynamite, right?
Albert wanted to give the money to young investigators.
He did, yes.
To get their career started.
He specified that it should be 1 to 3 investigators. So that was what I was talking about. No, he wanted this to be something that would support their work. Right. And yes, that's what it comes down to. He wanted it to go to people who could then support their future work, which is actually what a MacArthur Award ends up doing. And you can look at the MacArthur Award and see, that type of how that plays out. And there are others like that.
The problem
is, the Nobel, was not supposed to be given to a bunch of old folks, but that's the way it's played out.
I mean, when he made these prizes, there was very little, if any, federally supported research, right? It was all by wealthy people giving you money. So he wanted to change that, but I think they couldn't find young people to give it to, so it ended up morphing. Speaking of a MacArthur Prize,
and by the way, Charpentier and Doudna are not old. Certainly not by Nobel standards. They actually could use this to fund their future research, although I don't think either of them has a particular funding problem.
They don't need it, but they've been working for a while. It's not. I think he really wanted someone
They are definitely senior scientists. Yeah. He wanted it to go to somebody.
Speaking of MacArthur Awards, our good friend Nels Eldey just received one. Congratulations, Nels.
Yes, good job, Nels.
Which is a nice award. It's often called the Genius Award, right? Mm-hmm. So now we're in the company, here on Microbe TV, we're in the company of a genius.
Company of a genius, yes.
We have a paper today, which is really interesting. But before we do that, I wanted to, a very interesting article out in Morbidity Mortality Weekly Report, Progress Towards Poliomyelitis Eradication, Afghanistan, January 2019 to July 2020. So Afghanistan and Pakistan are the two last countries where wild-type poliovirus continues to circulate and cause paralysis. They are not the only countries with polio, unfortunately, because in many other countries, the vaccine is circulating. I shouldn't laugh, excuse me. The vaccine is circulating and causing polio. But wild-type polio, type 1, continues to circulate in Pakistan and Afghanistan. And unfortunately, so these countries saw a decline in the number of cases due to great immunization efforts. But since April of 2018, insurgent groups in Afghanistan have imposed bans on house-to-house vaccination. And so that was the main way that immunizers could get to many children, right? Go house to house. They would actually mark the door so they could know where to come back or not. But these have been banned by insurgents. Why, I don't know. But so that you would have to go to a healthcare facility, and that's very hard to do, of course. And then, of course, from March to June of this year, all the immunization campaigns were paused because of the pandemic. And as a consequence, the number of wild-type polio cases in Afghanistan have increased from 2018 to 2019. I mean, there are 29 cases in 2019. And then during 2020, 41 wild-type cases, but in addition, 69 cases of circulating vaccine-derived poliovirus type 2. Now, type 2 was declared eradicated in 2016, and so they've withdrawn the type 2 component of the vaccine, but the vaccine strains are excreted in the feces. They continue to circulate for a while. And they can cause outbreaks because they are revertants shed in the feces. And, if you stop immunizing, there's still some circulation. So there were outbreaks in any country where you let immunization drop. There were outbreaks of type 2 polio. And what would they do to quell them? They would go in with trivalent OPV or monovalent type 2 OPV, because OPV is the great way to stop an outbreak. And that would recedes the type 2 and 3. Yeah. Yeah. So
but this is where Raul's stuff is supposed to go.
Raul and Dino and Andrew McAdam and others have developed a new OPV2 strain. We've never talked about it. We should do it sometime, which they believe reverts less.
Right.
Okay. And there is talk about implementing this to replace OPV2, and we'll see. I mean, it's only been tested in a few hundred people, and there's nothing like a few million kids to test the real stability, the real genetic stability of a vaccine. But that may come into play.
The way out of this treadmill where you keep having these reemergence of vaccine-derived polio is, at least in the developed world, it was to switch over to the inactivated vaccine. Yeah. So when you get your polio vaccine, your kid goes and gets it. I got my oral vaccine when I was a kid, but now you take a kid to get a polio vaccine, it's a shot. And that's derived from the Salk vaccine. And that cannot produce polio. There's just that virus is completely inactivated. Yeah. Unfortunately, in a lot of the world, giving somebody a shot is just not feasible because you don't have folks. The nice thing about the oral vaccine is someone can take a brief lesson in how to give it, and you just put some drops on the kid's tongue and you're done. Yeah. Whereas a shot actually has to be delivered by somebody who has a little bit of medical training, and that's just too scarce. So
actually, the WHO recognized that not having OPV2 in the vaccine was a problem. So they said, okay, in Afghanistan, they said, we want you to do 1 dose of trivalent IPV at some point. But it's hard, as Alan said, there's a number here. Estimated 1 dose IPV coverage in 2019 in Afghanistan was 66%. It's not enough.
That's not going to do it.
It's not enough to protect the kids. So that's because it's hard, as Alan said, to inject. It's really hard. Anyway, this is a wonderful article.
More expensive, too.
It's more expensive. So this is a wonderful article. They have great numbers, and they give you a sense for what people are doing. House-to-house campaigns reaching millions of kids less than 5 years of age. But here's some sad part, and they have data saying that in 2019, in March, almost 500,000 children less than 5 years old, which is about 4% of the population in Afghanistan, were not accessible during the March National Immunization Day campaign. 500,000 kids they couldn't reach. And those kids are the ones who end up getting polio as it's circulating. In fact, even kids who get 1 or 2 doses, you need more. You need 3 doses or more. Even those children can get polio as well. The other things that are going on, and this is something that Amy likes, they do surveillance for acute flaccid paralysis. So they look at kids with 1 or more weakened limbs. It's called AFP. And then they say, okay, if we detect this, they say detection of 2 or more cases of AFP per 100,000 persons less than 15 years says that we're doing good surveillance. And then 80% of these have to have stool specimens taken and look for polio to see if it is in fact a polio case, because other viruses and other things can cause AFP. And then they also do sewage sampling. They have 23 sites in 11 provinces where they look for virus, and you can find it. When virus is circulating in people, you'll find it in the sewage because this is fecally excreted, of course.
That's how it spreads.
Yeah. And these type 1 strains, they can sequence their genomes and see that they go back and forth between Pakistan and Afghanistan. You can track them. And this type 2 cases that they're seeing is a relatively new outbreak. It started in Pakistan in 2019. It's spread to Afghanistan, and it's growing. So type 2 vaccine-associated polio there are more cases of that every year than wild-type polio now globally. There are a few hundred as opposed to maybe less than 100 type 1 cases. So that's kind of ironic, right? This is the vaccine that nearly eradicated polio globally, but now it's causing most of the polio. And
it's that last 1% that you just can't, I think, can't get rid of with the oral polio vaccine. No matter how you reformulate it or what have you, it's gonna, as long as you don't have the 99% coverage that we could get in the developed world, and as long as you're not switching over to IPV, this stuff is just going to drag on and on. And in a place like Afghanistan, the political situation, the logistical situation is such that it's just really, really hard. And of course, the pandemic has made all that worse.
So we'll see if Raul Andino, Andrew McAdams' new OPV2, if it's, I think it's going to be used at some point. We'll see if it's better. But, we know that IPV would work if we could get that to everyone. And, to solve the needle problem, I think it needs to be converted to a microneedle vaccine. These microneedle patches that you can put on skin with a Band-Aid. You should do some trials to see if that works, because that would get around needing to have needles and trained healthcare personnel and so forth.
The other thing about delivering an injected vaccine is if we could focus on building and actually accomplish building the infrastructure to do that to the whole world, whether through microneedle or just training people up or, just getting enough money mostly, but also people and training, everywhere in the world that we could deliver an injected vaccine, this would be a great platform with which we could deal with all the other vaccine preventable diseases that are, spreading in these areas like measles, mumps, rubella, et cetera, et cetera, et cetera. And that's all still going on, because any place you can't get needles in arms, you're going to have all those diseases spreading.
Well, there are measles outbreaks due to the pandemic.
Yeah,
measles numbers have increased tremendously.
Now,
one would think with the idea that we're going to vaccinate the whole world against SARS-CoV-2 and somehow manufacture it in India and various other places, that this infrastructure might be a consequence or could be repurposed.
Yes, this might finally be the kick that we needed to get this done. I hope.
We'll see. Now, one of the viruses that Amy works on also causes paralysis in kids. And, that's Enterovirus D68, which you have talked about on TWiV before, right?
Yes.
Yeah.
And we're supposed to have outbreaks every 2 years. And so this is the outbreak year, and it's supposed to start in August. So what's going on with that, Kathy Spindler? Amy, what's going on with that?
I'm just the sit-in. So AFM, there have been cases of AFM this year. There are more cases this year than there were last year, but there hasn't been the outbreak that was expected or anticipated, most likely due to the physical distancing and lockdown and people not sending their kids to daycare and nursery school, because it's really a disease that affects children under 6.
So the virus hasn't mutated, Amy?
No, the virus hasn't mutated. We have
no. The virus mutates all the time, right?
Well, I don't like the word mutates.
Yes,
but yes.
Right.
But yes, the polymerase makes errors all of the time, and 9/10 of those errors are probably lethal, where you don't get any productive infectious virus produced. But what remains mostly is probably silent.
Yeah.
So you
phenotypically, it hasn't changed.
You told me that the CDC is behind in their reporting of EV68, right, in the U.S.?
Yes. Yeah. So last month, Steve Oberste, who oversees the polio entero division, alluded to them being behind and only processing up to like the May, June samples. Oh. But I'll hear more about it on Tuesday at my AFM meeting.
So Rich Condi, I think you know Steve Oberste, right?
Yeah, he was a Flanagan guy, right?
He's a student with Flanagan.
Came out of Flanagan's lab at UF. I think, I overlapped with him. I probably was I think he was a PhD with Flanagan or a postdoc. I think a PhD.
Yeah,
I was there, probably went to his defense seminar.
I was on his defense committee, so I must have seen you.
Wow. Did you visit UF to do that?
Yeah, it was pre-TWiV, though. I don't know if I was talking to you.
Probably not. So, we knew each other on study section around the time I moved to UF.
Yeah.
So
what you're saying, Amy, is
I was probably scared of you, Vincent.
No. You were the scary guy. Come on. So, Amy, what you're saying is that maybe when the CDC catches up, we'll see a backlog of cases that actually are happening? What do you think?
I don't know that we're going to see a backlog. I think we'll see a few cases. I don't think we're going to see the 200 to 300 that we saw in 2018. The most probable explanation is that we'll see a few cases, then it'll decline again, it'll go back up. We'll probably now be on the odd year cycle.
Okay, so next year we'll have an outbreak.
And then because people aren't exposed, we might see an actual increase over the 200 to 300 that we saw before. So we might see closer to 500.
So, Amy, is there any explanation for this every other year behavior?
So not at the moment, there's not, but we have some ideas. One of them is the idea that there's a naive population of children and it builds up to a point where we can detect it because there's always a background level of AFM that's non-infectious or related to other enteroviruses such as EV71 and Coxsackie 16, which are more prevalent, especially in Asia and in the West Coast.
So the idea would be it takes 2 years to build up a sufficient density of naive people to propagate the infection.
Or even to detect it above the background numbers. Right. But also the definition of AFM has become very specific. So previous cases, probably if you did a retrospective study and you had MRIs, we might actually see more AFM than we thought. Because now, to fulfill that definition, you have to have an MRI that has lesions within the anterior dorsal horn of the spinal cord. So it's very, very specific in some respects.
And this alternate year observation doesn't go back like 50 years. This is a fairly recent thing, right? That we've actually been able to see that signal.
Yeah. So the virus isn't really, I guess the virus is about 50 years old. So it was initially isolated in 1962 from children. And then there was no detection, but it was also classified as a rhinovirus. Then it was reclassified. It's kind of like, we don't know what you are. But it's
what I'm getting at is,
but it's not until 2014 that we saw this.
It's probably been in humans for a very long time, but we're only noticing it because we've dealt with, say, polio, where presumably if you got Enterovirus D68 AFM in the past, in the 1950s, they would have said, oh, you've got polio.
Yeah, that's probably true. But the other thing is, a lot of cases probably were misdiagnosed.
Yeah.
They're still being misdiagnosed or missed because we do a BioFire multiplex PCR and the primers aren't really that specific. So you get grouped as enterorhino positive. And then when you present with a common cold, your mother just says, I told you it was a summer cold, and you go home and you resolve it. And it doesn't get really doesn't get refined further.
All right. So next year when there's the outbreak, we'll have you back to talk about it.
Yeah.
Yeah. Hopefully we'll have some data.
Yeah. All right.
I don't like to promise anything.
So we have a paper for you, which is all about SARS-CoV-2. And this is published in Science. It's a research article. And it's another one that I can't highlight. And if I do, I can't save the document.
I'm the same. Yeah, this is really annoying. If anybody at Science who's in their tech department is listening, you need to knock this off with the PDFs.
I did get some hints from people from the last time, and I tried them, but they didn't work. And Amy will attest that I tried them today. Anyway.
Yes, but what is interesting is the paper behind this paper, which is by basically the same group. You can highlight on.
Yes.
You can highlight it, really.
You can, yeah.
Okay.
Because I read that paper.
This one is called Inborn Errors of Type 1 Interferon Immunity in Patients with Life-Threatening COVID-19. And there are a lot of authors on this paper.
A lot of authors. I'm not sure I've ever seen this many authors except like on a physics paper.
And not only are there a lot of individual authors, but a lot of the authors are like the Imagine COVID group, the French COVID cohort study group, the COVID contact cohort, all these organizational, which would therefore be probably hundreds of people behind them. It is the author list and affiliations extend almost through the first 2 pages of the paper.
And there are consortia as well. the COVID storm clinicians, the French COVID cohort study group. So there are probably many, many more.
This was a big, big project.
But
For 600 patients.
There seems to be one first author, Xian Zhang, and 15 second co-authors, and a bunch of PIs at the end. And the last person is Jean-Laurent Casanova from Rockefeller University, who is known for doing these kinds of studies to say, do people who develop serious viral disease have SNPs, single nucleotide polymorphisms, or mutations that associate with disease? And I want to point out that also on this paper is Charles Rice, who got the Nobel Prize this week for hepatitis C virus. A lot of other names we know: Tom Maniatis, Florian Kramer, Adolfo Garcia Sastre, Michel Nussenzweig, and Trine Mogensen, who is a clinician in Denmark who I had on TWiV, and she talked about her study where she did a similar analysis with kids who got polio, because only 1 in 100 kids who are infected with poliovirus get paralysis. So she said, are there any mutations that associate with that? I guess a lot of these authors donated specimens, and that's why they're on the paper. Anyway, so the question here is, with SARS-CoV-2 infection, you see a big range of clinical presentations. Some people have no symptoms and other people die. And they say there are three things that influence this severity risk: increasing age, being male, and having underlying medical conditions. But even within these groups, there's still a big variability, clinical variability. So they want to know, he's established the COVID human genetic effort, and there's a website which you can go and look at that to try and see, in people with serious disease, do they have mutations in genes that are associated with that? And that's what this paper is about. They have 659 patients of various ancestries, aged between 1 month and 99 years of age, all of them hospitalized for life-threatening pneumonia as a consequence of SARS-CoV-2 infection. And they either sequenced the whole genome or did exome sequencing of...
which just blows my mind. Oh, yeah. 600 people, we just sequenced their genomes.
And they've got 500 and some odd controls, too.
Right. What a world.
Yeah.
It's Gattaca, dude.
Yes.
I need to rewatch that movie.
Yeah.
I never watched it. I'm sorry.
Yeah, me neither.
It's good. It's good.
Is it good?
Yes.
All right.
I'll rewatch it and confirm that. But
right.
My recollection is what I didn't, I'll probably watch it again and think, holy cow, this was more prescient than I thought.
So the title
Gattaca
is
before genome sequencing.
Is GATTACA.
Ethan Hawke? And,
it's definitely Ethan Hawke.
The title is the nucleotide sequence, right? GATTACA.
Right. Basically, this is a digression here, folks, but, the theme is that essentially when you're born, your genome is sequenced, and that's fundamental to your identity.
Yes.
Okay. And there's a lot of eugenics that goes on as well, because certain genomes aren't allowed. Okay. And it's a kind of a 1984 Brave New World type approach to this, where there's some person who's not playing by the rules. Okay. So,
well, I have to point out, and this is a digression, but I think this is so funny. When the book Jurassic Park came out, the idea there was that they got mosquitoes that had been trapped in amber from when dinosaurs were around. They got their DNA out and they sequenced it, right? And they put together a dinosaur genome. But there's a little bit of sequence in the book. And someone blasted it, and it's PBR 322, a cloning vector. So
good.
Michael Crichton, he couldn't just make up. He had to pull this out, right? Isn't that funny?
Yes.
That's great. And, I think we're approaching the time when your genome sequence is going to be part of your medical record. I can see that happening. Hopefully I live long enough to see that. Not that I necessarily want to see it. I just want to live that long.
You just want to live that long, right?
You could live another 20 years.
Oh, yeah. I got 20 years in me. I'm pushing for 30. I want to make 100.
You want to make 100?
Yeah. Yeah, I want to make 100. Then I can crash. That's okay.
Wow.
But I want to crash.
I can understand that. My grandmother made 100.
Yeah.
She was very, we
Really?
had
She hit 100?
Yeah.
Wow.
She hit 100. We have pictures of her. She had 100 crown. Like she had, I don't know, she traveled. She had boyfriends at various spots.
And then she died at 100.
She was like living the single life.
Great.
Sex in the City at 100. That was my grandmother.
All right. So
And this is why TWiV is 2 hours long. I hope
Yes.
your
Sorry.
dog is enjoying the walk.
sorry. Anyway,
Yeah,
so
I
what?
mean,
what
when she died at 101, the funny story is that she had booked herself on a vacation and she had never told my father or his sister. And the vacation people call up because they saw the obituary and they say, would you like your mother's $500 back? And they have no idea why these people are giving them $500. They're like, oh yeah, of course. But what did my mother sign up for? And they're like, oh, she signed up for a tour for the Navajo art exhibit in Buffalo in July.
You know,
Great.
those are the people whose genomes need to be sequenced to see why they have longevity, right? Not an easy
Yep.
question,
But
but
it's not all going to be the genome.
not all the genome, but they must have good sequences of immune,
Probably some of it. yes.
immune components and other things. Anyway, in this paper, they say, okay, we have these over 364 plus 295 genome sequences. So what's there? And they look in genes that they knew from previous studies are involved in life-threatening influenza, okay? People with mutations in genes for TLR3, toll-like receptor 3, which is a sensor of nucleic acids, leading to interferon responses. IRF7, which is a transcription protein involved in interferon response. These have been shown to be important for. They've been shown to be changed in people with serious influenza, and other loci, IRF9, another transcription factor, and a bunch of other genes, whose gene products are all involved in the interferon induction pathway. Is that fair, Amy? Say type 1 interferon induction pathway.
Yeah, I think that's fair.
Okay. So they say,
So
do these?
They're cherry picked. They're cherry picked
Yes, right.
to be really just about the fact that this disease causes a pneumonia. Influenza can cause a pneumonia. But
They did not just look at the whole genome and say, oh, there are these differences. Because if you do that, especially on a population of 600 and some odd diverse individuals, everybody's just different everywhere.
Very hard
So
to do.
they zoomed in on stuff that they thought might be involved.
yeah, I mean, there's a lot of caveats to what they did.
Yeah.
So one is that clearly from the physicians last week, we know that this pneumonia is very, very different than the pneumonia that influenza leads to. So one has to question whether or not this was, biologically relevant.
Well, they didn't
Are
listen
we
to
even looking in
TWiV.
the right place? Yeah,
Right.
Of course, that TWiV came long after they finished this work. So,
that's the problem.
and
Right.
even if it,
I didn't say.
Man, they're gonna listen to that TWiV and say, "Oh my God."
all right. So they
don't think
find.
Well, before that TWiV and Vincent's paper was on bioRxiv in March that demonstrated that SARS-CoV-2 doesn't induce type 1 interferon. It's sensitive, but it doesn't induce it. So one has to question whether or not any of this is relevant then. Because, yeah, you can be sensitive, but that if you're not inducing it, who cares?
Yeah, that's a good point. We'll come back to that. So they found mutations in these genes, IRF7, IFNAR, 113 patients with changes at 12 different loci in these genes that they looked for. 29 of these predicted to be loss of function, which means it would knock out the protein. They have others that don't seem to make any changes. And then the control group, 534 controls, these are people with either asymptomatic or mild SARS-CoV-2 infection. They only found one change in one gene of all these that they looked at. So that's kind of interesting. It seems to be enriched, right, in these very sick patients. Which doesn't mean it has anything to do with the severe disease. It's just that maybe they get sick with lots of respiratory infections. Not anymore, though. But then the key is experimentally, do these changes in these proteins matter? And so they have a variety of systems. They're mainly cell culture systems where they have cells that lack the genes and then they put the mutated gene in, or they overproduce the altered protein in cell lines, right? And they found that of these 113 variants, 24 of them hurt the cells that they were producing. They were deleterious. And so I don't need to go through them, but they're all, again, involved in the type 1 interferon pathway.
And this is, if people want to, not only is the paper open access, it's pretty short. It's like 4 pages, not counting the author list and the methods. So
the author list is the longest part, right?
I was going to say that it's like 2 pages out.
Actually, yeah, it's 2 pages and no figures.
Yeah.
It's a
the
pretty
figures are at the back of the page.
It is a very dense couple of pages, so it may take you a while. It certainly took me a while to grind through, and I'm sure I have not got all of this.
Yeah.
But one of the things that the main experiments are where they use reporters. They use reporters to look at interferon induction. So they're not actually measuring interferon. They're measuring, did you turn on the gene?
Yeah.
Right.
So the 23 patients had these 24 deleterious variants, okay? And these are in IRF7 and IFNAR1, which is the receptor for type 1 interferon. And I really very much like the abbreviation for autosomal dominant, which of course is Alan Dove.
Yes.
I just wanted to point that out.
Autosomal recessive is me. It's AR.
There you go.
And we have them both on TWiV. What a show.
There you go.
I don't like to be left out.
All right. So these patients have what seem to be deleterious mutations in these genes. Now, the question is, does it matter? Because these are cell culture experiments, as Amy said, may not mean anything. So they do what I consider to be the key experiments, but they don't go far enough. They take cells from patients, but only 2 patients, I think. And I appreciate that it's hard to get cells from patients, especially if they're dead, right? So you can't get those. And then others these are people from all over the world. But they have a few patients where they get T cells and plasmacytoid dendritic cells, which make a lot of interferon. And they say these dendritic cells from one patient did not produce any detectable type 1 or type 3 interferon in response to infection in culture with SARS-CoV-2. They never did that experiment with control cells, control patient cells.
Yes.
Maybe this is what happens in everyone, right?
But it's also known from Vincent's work and from others that when you take ALIs, human airway liquid interface cultures, and you infect them with SARS-CoV-2, there's no induction.
And yes, so that's Vineet Menachery, who was on TWiV a couple of times, right? Now he's at Galveston, right?
Galveston.
UTMB.
Yeah. He's at Galveston and from Ralph. And I'm sure Ralph has done the exact same experiment or similar experiments because he was a postdoc with Ralph. And, Ben Tenuver did a whole transcriptomics analysis, and he also doesn't see any or very low levels of interferon alpha or type 1 and type 3. And this is a mucosal surface, the respiratory tract. So it would be dominated theoretically by type 3, not type 1. And if you go to the paper that follows this paper by the same group that looks at autoantibodies against interferon, they look at and they identify autoantibodies against interferon omega versus type 1 or alpha. And they find, that these people who are interferon receptor or interferon who have these self antibodies against interferon are also sensitive. But, so we know that interferon protects them, but if the virus isn't stimulating any interferon, I'm not sure what the biological relevance of it is.
So that's your main objection, right? The virus has been shown by others that it doesn't induce interferon. So why would mutations in interferon pathways make any difference, right?
Right. It's the same argument I had when I used to say translation.
Yeah.
So people would come and say, oh, we found this inhibitor overproduced in cancer cells. And I would say, yeah, but the protein that phosphorylates it and inactivates it is also overexpressed. So the ratio is the same. So what's the difference?
The other experiment they do is they say, okay, let's look in the blood of these patients. Can we see interferon? They look for type 1 interferon. There are 13 different types of interferon alpha. And they find they have very low levels, right? But they do not look at their controls in the same way. They look at published results from other studies which say, yeah, these other studies, they have plenty of interferon and ours don't. But I think that's not a valid comparison. And
you'd like to compare stuff
system.
that's done in the same lab or with the same techniques. Yeah.
So I asked Amy, why was this published in Science? And she said, well, it's SARS-CoV-2.
Well, I mean, there's lots of caveats, right? So recently a Cell paper described that NSP1, NSP8 and 9, and NSP16 all turn off interferon production at various steps. Like they don't allow for mRNA splicing, they don't allow for translation, they don't allow for it to enter the ER because it's a secretory protein. They don't allow it to go through the secretory pathway. So there's hundreds of ways that, the virus can obviously inhibit interferon production, and it starts at the very beginning. But also, they pull out, they say TLR3 is really important. IRF7 is really important. If you go back to the literature and you look at what people have identified as the sensor for coronaviruses, that's MDA5. So either we're doing all the experiments in tissue culture completely wrong, or there's a disconnect between tissue culture and animals, which is most likely the right answer. But, why don't you don't pick out any of these proteins? And one of my last complaints is this is supposed to be an inflammatory response. Do these proteins now lead to turning on the inflammatory response instead of type 1 interferon? So are we mischaracterizing them and don't understand their full transcriptional profile?
It's an interesting point. Well, the authors are convinced that, at least in some patients, interferon deficiencies lead to severe COVID. And they say maybe we should treat them with interferon.
Yeah, I don't think Sam Perlman's going to like that idea.
Why is that?
Because he demonstrated that if you treat at least from midpoint to late infection with MERS, the treatment of interferon enhances pathogenicity. So it makes the disease more severe. So I'm not clear what early means. And
they do say early.
They
say early in the course, right? But that's the conundrum here, right? It's hard to get people.
Early in the course, these patients probably weren't hospitalized and that is coming up with a lot of the COVID-19 interventions because it's becoming clear this is kind of a biphasic disease where you get the viral phase and you can try and target the virus and you can try and target antiviral pathways there. And then there's this later phase that puts people on ventilators. And the virus is not the main story there. It's the immune system's response. And so the stuff that you do at that late phase is going to have to be different from the stuff you did at the early phase for the most part. I'm sure there are going to be some therapies that you can give all the way through. But yeah, this is the idea of giving interferon early and then maybe it's bad late. Do you really want to make that call? I mean, how long has he had the virus?
Well, it's a similar thing with steroids, right? You don't want to give it early.
Right.
But you do want to give it late, so you have to know what phase you're in. And
I think that this actually is very common, that you present with a mild infection and then you get severe disease later. Look at EV68, kids present with a cold. Mom says, no need to do anything. You have a cold. And then 10 days later, they have, some kind of limb weakness. Yeah. So here's the question. You have 40 to 60% asymptomatic people? That asymptomatic is early infection.
That's right.
By the time that you go and you say, I can't breathe so well, you're not really early in infection.
No, you can't give them interferon then. I agree. So Amy, the idea here is that the virus is inhibiting interferon, right? But still, in a large fraction of patients, they can still clear the infection, and it's only in a certain cohort that it becomes serious, and we don't know why still, right?
Yeah.
Right.
But I think research-wise, we have focused very heavily on every viral infection can be cleared by type 1 interferon to some degree. And I'm not sure that is correct. I think that that's our focus. I think that there are other early innate immune responses that we haven't well characterized because they're harder to find.
And it's important to note, even if we take this paper completely at face value and say, okay, let's say these mutations are involved in the pathogenesis, or some mutations linked to these are involved in the pathogenesis of the course of the disease, it is still a minority of the seriously sick patients that they've accounted for.
That's right.
It's a couple of dozen out of their 659 patient cohort.
Yep.
So there are a lot of people, the majority of people who end up severely ill with COVID-19, we don't get a genetic explanation from this paper.
So this is 3% of their cohort.
Right.
Number one. Number two, I don't understand how this would factor into the clinical observations that when you're older and over 65, that this is going to explain why you have more severe disease. Like,
that's right. It doesn't explain age dependency.
Just turn on then?
No, that's right. It doesn't explain age dependency. No.
Right.
Or male predominance, right?
Right. No, and they weren't trying to.
I
think they were looking for stuff that would be likely a minority of the patients, but are there particular alleles that are associated with really bad outcomes differentially? And they may have found some, but we don't know exactly why.
Well, they have a database now.
Right.
Yeah.
That can be mined for other things. It may not be big enough, but my guess is it's growing.
Right.
I think in their sequences, there are other things that may be very informative. So this is not this is not the end of the story by any means. And there are only 659 patients. A lot more people out there.
Yeah.
Well, it's also interesting.
Which is approximately the same as the number of authors on the paper. So
it's also interesting to contemplate, is that 3% what ethnicity or race or whatever you want to call it are they? Because here, at least in the United States, it seems to be hitting, a disparity, like an African-American population, a Latin population. So are these all within one of those groups, or are these, randomly throughout?
I think they might have commented on that. I don't buy the race issue as having a genetic component. I just on face value, the genes that determine race are not going to be representative of the diversity within the population. When you say African American, African is the most diverse gene pool. And granted, most African Americans are from particular regions of Africa, but it's still a tremendously outbred, genetically diverse population. And so I don't think that I don't think race is going to end up being a genetic.
No, I think it's as Bob Fullylove said. It's sociological roots.
It is, but you can never discount it. Right?
It is. I mean, it's a good question, Amy. That they don't mention in the paper if there was a particular ethnic group, and there are ethnicities where you do have a population of people who are very closely, related in some way, and you might be able. Classically, genetics is done in families or in groups of families that have shared a common heritage, and so that kind of thing can be very informative.
I mean, it kind of almost makes you want to do this study in like Iceland, where they have 50 generations,
very
of
little,
genetic,
and not a whole lot of outbreeding.
Yeah.
I was very excited about this because I know we've done some of his other work on TWiV, but I'm less excited after having analyzed it. Okay, let's do some email.
It is a cool paper, and I do think that something of this scope is appropriate to publish in Science, especially as a relatively brief report. And it's great that they put together this database. And I think that's really kind of a big point of the paper is we're doing this and here's the database and let's roll with this. And as it accumulates data, people are going to find other things in those data.
Without a doubt, this study has to be done. This is how you get at it, right?
Yes.
I just don't think we have the answer.
I don't think we can conclude much from the data as they stand, but I'm glad that they did this, and I'm glad that that's published now.
Well, I'm not sure that this study is, warranted the press that like the NIH pushed forward for it.
What did they say?
How it was pushed forward by the NIH as like, oh, look, this is this is one of the magic bullets.
Right.
I'm not sure that delivered.
That was a bit over the top. Yeah.
All right, let's do some email. Rich, can you take that first one?
Sure. Jenny writes, why can't we have cheap, rapid result antigen test like this in the U.S.? And she links to the site from a British company called Alpha Labs that is advertising a rapid COVID-19, wow, that's a redundancy, rapid COVID-19 edge antigen test. That we've discussed these before. These are the tests that use, that test for the presence of viral proteins in your serum. Using the viral proteins in your serum, that's what they mean by antigens. Using antibodies that are fixed to a piece of paper or some sort of substrate, and it works kind of like a pregnancy test. And the fact, this particular one, if you look at their site, results in 15 minutes. That's great. For 55 pounds plus VAT.
It's a bit much.
That's a bit much. Oh, what's a pound? I was just about to do the conversion. What's a pound worth nowadays?
Oh, it's well over $1, I'm sure, right?
Yes. About
or
20,
about
30.
there, something like that. At any rate, that's, as tests go, expensive.
Yes.
And I would say that there are antigen tests in abundance.
That is $71.52 US.
Plus tax.
Plus tax.
Plus tax. I would say test-wise, that's expensive.
Yeah.
There are a number of antigen tests out there of varying qualities, varying speeds and costs. And I think this is a market that is sort of, I wouldn't say in turmoil, but it needs to settle out a little bit until we can set the right standards, do the right deployment, have the right field testing of these things and settle on the tests that are right. But there is, I would say, a fairly serious effort afoot, not only in the U.S., but elsewhere to identify the appropriate antigen test to deploy. And I hope we get there sooner rather than later.
And it needs to be an order of magnitude cheaper.
Yeah, absolutely.
But
It should be free.
Well, okay, yes, but the actual expense of it, somebody's going to be paying. It should be free to the people receiving the test when they show up for work, certainly, or when they show up for school, or what have you. But the cost rather than the price should be in the $5 range.
I mean, Michael Minna is pushing this, and others in the U.S. will get there. I don't know when.
Before the outbreak ends?
Well,
before we've got a good vaccine in everybody's arm.
Well, you said, Amy, it's going to be 3 to 5 years still. So yeah, there's plenty of time.
Sure.
Well, yeah. I mean, I don't see where we're still at, like, I think 50,000 cases a day being diagnosed.
In the U.S.,
I think we just went up.
I will bet that there is a really good, relatively cheap antigen test. This is the easy bet within a year. Okay. That sounds like a long time, but if we're talking about, it could be that's still quite relevant a year from now.
Yeah.
Okay.
Yeah. But my point is that's 2 years into a pandemic.
Oh yeah. Sure.
I mean, that's kind of unacceptable.
Yep. I totally agree.
And when you do that, even if you argue that it should be $1 per person per test. If you're a family of 4, that's about $1,800 a year.
Yeah, that's interesting. Yes, $1 a test is, in fact, in many ways, not cheap enough.
Yeah,
exactly. So,
Yeah, I think if the cost has to be borne by the individual receiving the test, then that's the root problem there. But if you're going to be doing these things daily, I mean, there's the whole issue of how do you just support that on an ongoing basis.
I think, Rich, by the end of the year would be my guess for these rapid antigen tests.
The end of this year?
Yep.
I think
so.
that's realistic.
10 weeks. So. 10 weeks from now.
I don't know if they're going to be $5 by the end of the year, but
no,
I
don't know about the price, but
we could probably be in the low double digits, $10 range. Something that could be deployed on a much wider scale than the PCR test.
Amy, can you
in
take
10 weeks?
I think so. Yes.
Hey, I'm being optimistic.
And at $10 a test.
Maybe in the vicinity of $10 a test. I mean, we're down to $77 a test with these guys, and there are many other companies working on this that want to deal in volume and that have tests basically ready to go.
So let's just think about this logically. So Vincent, you come 3 days a week. That would cost $30 for you to come to Columbia, plus the astronomical cost to come to cross the bridge and the tolls and the gas. So now we're talking your, $65 a week to spend commuting is now going to be $150 to $175 a week to commute.
Can I have a raise?
When I get another grant, sure.
I'd
be happy to.
If you implement it as daily testing and it's still $10+ a test, then that's going to be a problem. Yes. If you implement it as weekly testing, you're still light years ahead of where we are now, where most people have never been tested, and even people who are working the cash register at the supermarket have probably never been tested. Then yeah, $10 a test is going to be a huge improvement.
But $10 still, if you're working the cashier as the supermarket,
going to be, right?
Is still buying you with chicken and yes. potatoes and vegetables 3 days worth of eating.
If the supermarket has to pay for the $10 and not out of your paycheck, then that is going to make a big difference, and you're being tested weekly, and then you know when somebody pops up positive, and you can take action sooner.
I think the businesses should pay for it, right? If they want to have,
I think the government should pay for it.
Well, the government should pay for it, but
that's true. Yes,
realistically, the businesses.
And we're talking here about deploying these things in a broad scale. I would also say that this is the kind of thing that can be used in a more focused approach as well. I've been doing some of these chat with a virologist things that come up with very interesting questions. And an interesting question from a school teacher is, so I've got a positive kid who goes home. Now what? Okay. And in areas in particular that are disadvantaged areas, you may have a lot of people living under cramped circumstances. That kid's probably not going to, really effectively isolate for an appropriate period of time. And one of my answers to that was, wouldn't it be nice if, in terms of the SIBs coming back to school, you couldn't test everybody? All right. That's a circumstance, a more focused circumstance where I can see this kind of thing being used.
Okay. Amy, can you take the next one?
Sure. So Dave writes, y'all make fun of Dr. Fauci that CT are not available. I guess he means CT values for...
Yeah, CT values for the PCR test, quantitation on the PCR test.
Yeah. Let me challenge you further. If I am not mistaken, most quantitative PCR SARS-CoV-2 tests approved by WHO, CDC, FDA use 2 primer pairs. I do not recall this ever being discussed. As you know, each qPCR primer pair, it's irrelevant of whether or not it's a quantitative primer PCR. It's just an inherent characteristic of primers. Is unique with its own CT sensitivity. Please explain why you would be satisfied with a single CT value. Also comment on the best primer pairs for qPCR for SARS-CoV-2. Furthermore, please comment on efficiency of single versus double primer pair tests with regards to detecting infectious patients. Thank you. Enjoy the show. I enjoy the show, Dave. So there's multiple things in this email that come to mind. So first, every primer pair, no matter what kind of PCR you use, has their own sensitivity, and that's depicted by the annealing temperature and various other characteristics. So you want, if you're going to use 2 primer pairs in one reaction, you want primer pairs that have very close or similar, or if not identical, annealing temperatures. When qPCR first, or real-time PCR first was done, we used to take plasmids that encoded the gene and did a titration for looking at when you saw the fluorescence increase, because the marker for a fluorescence of cyber green is binding to every single DNA molecule. And then you take a background, you subtract out the background, and that's how you get the curve of fluorescence. So we used to take plasmids, quantitate them in moles, go through the whole PCR, and then you could know exactly what you had and how much you have. So so please explain why you'd be satisfied with a single value. Well, actually, I'm not satisfied with a single value, but that seems to be the way the tests have been designed. And they seem to, generally, they use 2 primer pairs, one within the polymerase, because that's obviously the most conserved region. And when you don't know what you're looking for exactly, that's very easy to target. And then another primer pair that sits in the 3' end of the genome, which usually sits within N protein or the subgenomic mRNA. And this is a problem because you are also amplifying the subgenomic mRNA and not just the genome. So I'm not actually happy with 1 primer pair, because if you're just using primers to N, you could get false positives. So the best primer pairs are disputable. It depends on your machine. It depends on what you're using, about what temperature you're using, and various things. The most common primer pairs are, as I said, within N and the polymerase.
So Amy, can I ask you, when you do the 2 primer pairs, you get a single CT value from that reaction, right? Yeah,
from the way that they're doing them now, you get a single CT value.
So, Daniel Griffin always says, I want to see the CT, and that would be an average of the 2 primer pairs, right? By definition.
You try to take primer pairs that have the same.
Okay.
So for instance, if you look at the Broad website, they have 2 primer pairs. One that sits in ORF-A1b, which is targeting the polymerase, and one that sits in Spike, which is part of the subgenomic. They must have similar annealing temperatures, but they have different sensitivities. So I believe that the one that sits in ORF1A and B can recognize 10 copies per microliter, and the other one recognizes 100 copies per microliter. So you get an average value.
Which is appropriate, because the one for Spike is going to be sensitive to messenger RNA, which is going to be more abundant than, or subgenomic messenger RNA, which is going to be more abundant than the messenger RNA for polymerase, correct?
Yes. But ideally, you should be doing them in separate reactions, which would be twice the price, because you have 4 combinations, right? You have plus minus, minus minus, minus plus, and plus plus. And if you are plus plus, then you would know that you have full length virus, because you're detecting both one at the 5' end of the genome and one at the 3' end of the genome. So the probability of you having a full length copy of the genome is greatly enhanced. But, you have to, in order to understand that, you have to understand something about the virus, right? And I'm not guaranteeing that all of these things were done understanding the biology of the virus versus how we could rapidly identify infectious patients in the middle of an outbreak. So you have to compromise somewhere.
Got it. All right. Anything else on that one? That's a good.
I think that's fine. I mean,
that's a good, thank you, Amy. That's a good summary of the details of the test. I appreciate that.
Yeah. It's what happens when you're an egghead.
You don't think you're an egghead?
No, but obviously
it's not
I'm
a bad
delusional.
Thing. It's not a
That's
bad,
what I've learned through this podcast is I'm delusional. No. I'm the queen of the eggheads.
We are all eggheads.
Egghead is good. Means it's like geek and nerd. It's all good. Yeah. Alan, could you take the next one?
Sure. Vincent writes, hi, Dr. Rack. Consider this email question for upcoming TWiV broadcast. Hello, TWiV crew. This is Vincent Racaniello, the Lesser, Vincent's doppelganger. So we heard from this guy a while ago, didn't we?
Yes.
There's another Vincent Racaniello who's out there.
Geez, did we discuss this before? Are there more than two?
Well, there
Isn't one enough?
Excuse me?
I love you.
Oh my gosh. There are over 150 Vincent Racaniellos in the U.S.
Wow.
There are a few in the New York area. This one is up in Westchester, and he actually encountered me years ago. He does some legal work for Columbia. And then there's another Vincent Racaniello out on Long Island. So there are multiple, not huge numbers, but they're out there.
Okay.
This happens with many names, maybe even most names. I actually have a disambiguation page on my website for the other Alan Doves who are online. Recently I heard a guest on a popular midday radio show about health and nutrition claim that flu vaccinations can both make one susceptible to respiratory illnesses and increase one's risk of contracting COVID-19. Being a seasoned TWiV listener, my BS meter immediately went into the red, as did my temper. I found myself yelling at the radio. I have to admit it had a cathartic effect, perhaps more so since I count myself among the rarefied few who can adopt that expression as their own, literally. Yes, he is indeed yelling. A quick internet search revealed that the association with respiratory illness stems from a 2012 study, the results of which could not be replicated, while the COVID-19 risk association was from a 2020 study that under peer review was found to be incorrect. And in what dark recess of the internet did I have to search to find this debunking trove? The CDC website itself, and provides a link. Since this is the time of year when many people begin to get their flu shots, could you please, as a public service, explain yet again why it is vitally important to obtain a flu shot this year and how they are both safe and efficacious? Thank you for all that you do. Sending you my grumpy best, Vincent Racaniello. Thank you, Vincent Racaniello. So this little chestnut has been circulating ever since that 2012 paper came out. We've talked about this on TWiV before, that found some association of people who got their flu shot were more likely in that particular study group to get respiratory disease. And then you look in a bigger data set, you find this is just completely spurious, bogus. Unfortunately, once anything like that sees the light of day, the anti-vaccine nuts will never, ever, ever let it go. They will be citing this paper until the heat death of the universe. Because it endorses their worldview, and that's all they really needed, and so that's going to go on forever. And then this 2020 study again, I'm sure that's going to live on in infamy as well. But yes, you can go, despite current management at the CDC not necessarily doing a lot of things quite right, you can still go to the CDC website. They haven't quite deleted the whole thing, and find useful information. In terms of flu shots, yes, absolutely everybody should go out and get their flu shot this year of all years. You really don't want to get the flu this year. And if you do get the flu, you really don't want to have a bad time of it, because this is a terrible, terrible time to be in the hospital with respiratory illness. And we've got a vaccine against the flu. It's not a perfect vaccine, but it's sure as heck better than not getting one. I got mine last week. It was the Sanofi quadrivalent standard dose. I sat down there, went to the town flu clinic. The nurse who was giving it looked at my date of birth. She said, "Oh, you're a young one. You got the regular vaccine." Oh, great. Thanks.
Did you get yours,
Haven't
Rich?
felt that good since the last time I got carded, which was quite a while ago.
Rich, did you get yours?
Yeah. Not yet. It's on my radar.
Okay.
I got a ping from CVS recently advertising that I can go down there and get my flu shot for free, which I will do. I need to do that. One of the things I appreciate a couple of things about this email. Okay. One is his description of his BS meter. Okay. If we can do anything on this show, we can give everybody a BS meter. All right. So that's important. And I really enjoy his description of how once his BS meter went off, he tracked this sucker down.
Yes.
And that's what people need to be doing. You know, we don't, there's a lot of misinformation out there, but we don't have to fall for it. Okay. You can do this yourself. Anybody can do this. And I'm pleased that there is this CDC site that debunks the misconception. That's good.
I would expect nothing less from Vincent Racaniello.
Yes. Yeah. And Vincent, Amy, have you gotten your flu shots yet?
We are.
Can I take the 5th?
We're going to go next week. I have to get the high dose.
The old person why we have
it's okay if you haven't had it yet, but people
next
need
week.
to get this.
No, we've been talking about it, but I have. What did you say? The old man?
No, I said you had to get the old person one. Rod called it the old person one. Remember? He sent us the whole email. Sure. We have to call Denise. We have to do everything.
See my ID I got here? Every year you have to have it to get one of these dots on your ID. Otherwise, you can't walk around the hospital without a mask. Of course, this year it's moved.
This year, nobody can walk around the hospital without a mask.
But that shows the previous years, and I've gotten them, so I will get it. No worries.
Well, we have to go anyway because in order to use the ABSL3 facility, he's listed as the person if I'm not available for them to call. So he has to have it in order to take care of whatever needs to be taken care of if I ever were to go on vacation.
What is an ABSL3 facility?
It's where you do animal experiments for biosafety level 3 pathogens like SARS-CoV-2. So it's very secure. Nobody looked at my helmet?
Yeah.
My lovely helmet picture?
Amy.
That my mother thinks is the Halloween costume of Halloween costumes?
So you have to get a respirator, which you wear. It's not like a BSL-4 where they pump oxygen into your... But you have a helmet with a filter unit on it, right? And Amy ordered one. It was like $2,500. Took 2 months to get here. It's amazing.
It's an expensive Halloween costume.
Well, that's what I said to my mom when she said, "Oh, look at how cute you look! It's a great Halloween costume." And I said, "Mom, for $2,500, it needs to be better than a cute Halloween costume."
But you will be here. She is in the helmet, right? For those of you watching.
Nice.
Nice.
That's
now,
good.
We didn't put the filter unit on, so it was fogging up very quickly, right?
Yeah. So I said, I can't use this thing. I can't see through it.
So you need to have a flu vaccine to go in there, right?
Useless.
Yeah. Okay. All right. The next one is from Gordon. I thoroughly enjoyed your interview 666 on the exciting prospects of far UVC light, far UVC, right? A few various thoughts. Most UV items found online are junk UVC devices, and to the extent they are effective, are also unsafe. Yeah. 2, there are companies which do sell effective handheld UVC devices. I don't know if they're far UVC, such as this one.
That's an important distinction.
Yes.
And he gives a link to one here. Maybe you guys can take a look and see if it is far UV. Because I found one randomly. It wasn't far. It was just UVC. This requires proper skin and eye protection, so safe use around the home is a concern. Despite the good amount of power, I find it takes a couple of minutes with this for me to fully scan our newspaper front page.
This is 253.7.
Yeah.
Wasn't it what was
220,
it? 220,
I think.
220?
Yeah. Yeah.
So that's not right.
Oh. The Columbia lab has also studied the use of a far UV in a leaky fiber optic setup with a catheter to keep the wound clean. This was mentioned to me by Dr. Bonanno when I asked if there might be a way to introduce far UV into the upper airway and respiratory tract. 4 product literature from Ushio, Japan regarding CARE 222 far UV lamps indicated it takes several minutes to deactivate airborne pathogens. So the name of that CARE 222 kind of suggests that that's the wavelength, but we don't know.
Yeah, far UVC is 207 to 222.
Mm-hmm.
And I'll
that's
look
at
up
the
this.
very end.
Right. This seems comparable to other technology for use in HVAC, the needlepoint bipolar ionization systems from GPS Incorporated, which generates hydroxyls and neutralizes airborne pathogens. The literature mentions having installed this in the White House. I will reserve comment.
It's good if
not
they
working.
installed it in the White House HVAC system, because there's somebody going around coughing up a bunch of SARS-CoV-2 there right now.
Well, it's not been working since Mitch McConnell has declared that he has not been to the White House since August because they don't know what they're doing. It's too dangerous for him.
That's what he said?
Yeah.
Yep.
Then he says test results for SARS-CoV-2 for this company, Innovative Bioanalysis. Oh, this is the company who tested some other company. SARS-CoV-2 neutralization by needlepoint bipolar ionization. So they have pictures of cells in culture, growth curves, and everything. It's the real data. Yeah, it's just demonstrating that this will inactivate infectivity. Thanks very much for being such a welcome source of healthy information, Gordon. Thank you very much. All right, Rich, you are next.
Melissa writes, Dear TWiV gang, I'm writing from Baltimore, New York, out on the island.
Belmore, New York.
Belmore, New York. Sorry. I'm an old guy. Out on the island, not too far from Jones Beach, where it is currently 24C and overcast. I just wanted to start off by saying how happy I am to have found TWiV a few months ago. It's allowed me to be happily reconnected to my former scientific life. I earned a PhD in biological sciences at our dear Columbia University in 2007, where I worked in the lab of Carol Privy's. Excellent. I had to leave research during my postdoc in 2010 because my daughter was born with medical issues, and I miss it every day. I love tuning into TWiV and feeling at home again. I love the Journal Club feel of the podcast, as it was one of my favorite parts of being in the lab. However, I don't miss giving lab meetings. Emoji. I am currently on a new career path that is completely different from what I did in my former life. Thankfully, this new path still uses the analytical part of my brain that I developed after nearly 15 years of lab work. I'm finishing a training program to become a certified reading specialist for children with dyslexia and other language-based reading and writing disabilities. My training is in the Orton-Gillingham approach. This is considered the gold standard methodology for remediating reading difficulties. It involves an explicit, systematic, and multisensory approach to teaching the English language. Trust me, to teach the English language or learn it, you need as many approaches as you can get. It instructs kids how to break the code and apply systematic rules to reading. These rules are needed to cement the neurological pathways in their brains and to make reading fluent and automatic. My writing my Ortho Gillingham lesson plan feels very much like when I used to design experiments, as there is much attention to data analysis and using data to guide the future steps. It is a highly diagnostic and prescriptive approach. Lest you believe that the current epidemic of science denying is limited to climate change and COVID-19, rest assured, the science of reading is equally maligned and disregarded by the masses, despite strong evidence to support the techniques it proposes. Evidence in support of the techniques it proposes. I don't understand how or when science became the enemy in this world. A few things mentioned last week really resonated with me. I loved Alan Dove's mild rant on the lack of curation with NCPI PubMed. Having worked in a P53 lab for my PhD, I understand the pain of having to weed through dozens of questionable publications with every PubMed search. At the time of my thesis defense in 2007, there were approximately 40,000 publications with P53 in the title. A quick search today reveals over 102,000 publications. Yikes. I remember the mythical rule of thumb that was circulating around my lab at the time when I wrote my thesis, that my advisor wouldn't even read your draft unless you cited 1% of the publications, or 400 articles in my thesis. I can't imagine having to cite over 1,000 articles today. I enjoyed a recent discussion with David Brenner regarding the use of far UVC light as a possibly continuous antimicrobial treatment in public spaces. One question that came to mind is that if there are any any possibility for microbes to gain resistance to the widespread use of far UVC. Could there be a selection pressure to circumvent the mechanisms that lead to microbial death and allow replication despite DNA damage? Also, because the far UVC will kill all types of microbes, both pathogenic and non-pathogenic, could there be other unintended consequences to the ecological balance with such a widespread approach? I feel like I may have better known how to answer these questions when I was still in the lab, but these days the information I need is in the far recesses of my mind. Thank you so much for your always intelligent and fun podcast. I have learned so much and try to pass it on to my non-scientific friends and family whenever I can. I have held several informal seminars in my kitchen to explain PCR versus lick a stick testing for SARS-CoV-2. I think my husband is getting sick of hearing me say CT value. Stay well, Melissa. P.S. I was also today years old when I learned that the UV light in the cell culture hood isn't blue. Emoji. Okay. So let's see here. First of all, I'm very interested to hear your, I was going to say rant, but it's not really a rant, description of teaching the English language. I have this conversation with my grandchildren all the time, ages 12 and 9, who are, they're pretty good writers and spellers at this point, and good enough to realize how crazy it is out there in the English language world, and I'm sure other languages as well. So it would be interesting to know what the methods are to deal with what in many cases looks like no rules at all. At any rate, that's for another podcast, I suppose. Far UVC light.
Resistance.
My sense is that if you zap something hard enough, they're screwed. Okay. Yes, there are mechanisms to repair in bacteria to repair UV damage and radiation damage and in mammalian cells. But I would suspect that you're unlikely at some level of UV dose, you're unlikely to be able to craft resistance that can deal with it. It feels to me almost like, multi-drug therapy for a disease. If there are enough hits from radiation, it's going to be hard to repair it. That's my feeling about that. And in terms of, others may have other reactions, in terms of other environmental damage, it doesn't bother me. I think that, we're talking about disinfecting surfaces in a fairly confined space that's a very small fraction of the whole world. And the minute you turn your back, it's going to be reinvaded by all sorts of critters. So I'm not worried about that either.
In general, from what I've seen, physics is really hard to develop resistance against. So UVC, the mechanisms that a virus in particular would have to get in order to overcome UVC, it boggles the mind. I mean, because the virus is dependent on the cellular machinery, so you're going to have the human epithelial cells in the lung evolve something that's going to help the virus out of dealing with these mutations that have been induced by these hits from the light source. The reason we see resistance against things like antibiotics is because the antibiotics have to be non-lethal to us. So we know cells can survive the antibiotics. If you can throw a threat at a microbe that can be completely lethal to all life, like UVC or, a temperature of 300 degrees Celsius or a nuclear blast or what have you, then there's not going to be any resistance against that because there's just no mechanism there. Now there are bacteria. There's one very famous one, at least in microbe circles, that it's called Deinococcus radiodurans. And it is the most radiation resistant life form ever discovered. You can hit this thing with gamma radiation until the beaker turns brown, and then it grows. And it's got its own mechanisms to do that, but it's a very specialized organism, and you're never going to get infected with it because it's not that kind of bug. And I don't think we're going to see any bacteria pick up those kinds of mechanisms because there's a huge evolutionary cost to be that radiation resistant. It's hard to be Deinococcus radiodurans. And it's not just something that you can bolt on to an existing organism. So I'm not at all worried about resistance arising to something like UVC light.
Amy.
Vincent, is
Yes.
Carol Privy's still around?
Mm-hmm.
She working?
Yeah, still has a lab. Yeah.
Is she in the medical school?
No, she's downtown at the Morningside campus. Yeah.
Okay, because I had, at least disciplinary overlap with her. She's a small DNA virus type, right?
Yeah, definitely. She is still there, and I've seen her on committees and so forth. I believe she still has a lab. She teaches undergrads. Yep.
Kathy has a little note here that says, lucky for Melissa to have a chance to work with Carol Privy's, one of the women in small DNA viruses ahead of me in her career, says.
Amy, can you take the next one?
Sure. Anonymous writes, Dear TWiV team, a friend of mine recommended TWiV back in February, and I've been listening ever since. I live in Hong Kong, so I listened to episode 665, where you discussed the paper on super spreading events with interest. I'd like to fill in some details on why we've been pretty successful in controlling the spread of SARS-CoV-2, despite being a highly densely populated city and without having to go into full lockdown. We did close the schools and a lot of public venues, but shops stayed opened, and restaurants were still allowed to see customers before 5 p.m. We adopted near universal masking as soon as we heard about the virus in Wuhan back in February without being told. Wow. Our previous experience with SARS made it impossible for anyone to be an anti-masker, no matter how they distrusted the government. In fact, at the time, the government was telling civil servants not to wear masks to conserve the supply, and was still defending the ban on masks at public gathering in court, intended to stop protesters from hiding their faces. People masked up anyway, and political activists organized to distribute them to low-income and elderly residents. And here's a link to the Atlantic article, I guess, that talks about that. Travel was shut down in March. Only Hong Kong residents were allowed to enter, and everyone arriving had to be quarantined for 14 days. People had to spit in a tube and get tested at the airport. In fact, medical workers went on strike in early February to demand a complete border closure and better supplies of PPE because they thought the government wasn't being aggressive enough. After weeks with virtually no cases, we had a second spike in July because it returned flight crew, sailors, and corporate executives, among others, who were exempt from quarantine requirements, so the virus got back in. And I guess here is their link to their newspaper article. 3, Tetris, we have a pretty robust public health infrastructure. Everyone who tests positive is hospitalized or put in a temporary medical facility regardless of symptoms. Their close contacts are also tested and required to live in a quarantine facility until their tests come back. That is why all infection chains terminate once they got to family clusters. The unpopular political leadership has mostly had the sense to not turn testing into a partisan issue. That is until recently, when Chief Executive Carrie Lam accused medical professionals who criticized the government's mass PCR testing scheme on grounds of privacy and usefulness of being politically motivated and conspiring to smear the government. I guess this is the other link to that article. I suppose if there's any lesson to being drawn from Hong Kong, it is that decent public health institutions combined with strong community spirit can make up for lackluster political leadership. Also, while I'm glad that testing is finally gaining traction in the US, I feel like the trace and isolate part still don't get enough attention. Our testing capacity actually was lower than Macau, a city with 1/10 of our population, but we sure did trace and isolate people. Anyway, love the show. Stay grumpy, Anonymous. So I think that here there's a lot of issues that distinguish the United States from other parts of the world. And many of their Anonymous's points are points that I'm sure you've all talked about, and still I cannot wrap my head around. So we don't really have problems putting seatbelts on in the car or wearing helmets when you bike. Although I was recently told by someone that like Pennsylvania and Ohio don't have bike helmet rules, but the majority of people you probably see biking around in Philadelphia and Cincinnati are all wearing helmets. So I don't really understand why a mask is actually any different than wearing a helmet or using your seatbelt. I think that we've shown that the travel shutdown that occurred here in the United States didn't really have an effect on the outbreak because it was already here. So I think the honest answer is that we're not honest because we think that if you test positive, it's like a stigma and you're not macho or something or other. So you shouldn't quarantine. You should go out and infect as many people as possible. And the public health infrastructure. So that's a big problem here in the United States. It's not independent from the government, and it should be actually. And I think that was highlighted in actually Varmus's editorial in the Times calling out to ignore the CDC, and also recently by the former CDC head of the CDC who's come out and told Redfield he needs to stand up and resign because he's made significant errors. But I don't, so from our experience, at least for EV68 and some other viruses, where we do interact with the CDC, I can say we have seen a change in how those agents are dealt with. And I think Vincent will attest to the fact that we have a neighbor across the hall or across the street who will also agree that it's not a SARS-CoV-2 issue per se. It's the whole public health infrastructure.
This pandemic arrived at a time when our government was uniquely already broken, like worse than usual.
Yeah. But the CDC has gotten more broken, and it's not a SARS-CoV-2 broken specificity. I think that our neighbor across the street would say that it is no longer a tightly run ship. No. Would you disagree with that, Vincent?
No, I agree with that. Yeah.
And I can tell you, like, when we ask for things for EV68, it takes months to get a response. And the response generally is like, I don't really know.
Now you're not going to get
I
anything.
couldn't recover a virus or something or other.
My wife and I were having this conversation this morning about this politicization in particular of mask wearing. And peripheral issues. And we were supposing that the leadership changes as a result of the election, and wondering if new leadership can fix the problem. And I'm thinking the damage is already done. It's so deeply political at this point that those who are so into this idea that the choice of wearing a mask is an expression of freedom or whatever, it's going to be really hard to change that culture, no matter how positive you are. I hope the effort is made. I hope the effort can be made. But
so actually, one of my students who's home from school because Stanford didn't enroll anybody, and she's now taking Stanford on her living room couch, as she likes to say. So she has family and friends in Upper New Jersey who feel that wearing a mask is a violation of their civil liberties and a violation of the Second Amendment. And I'm not really sure, but okay.
The Second Amendment.
Yeah. So the Second Amendment, which is you can hold a firearm, is really a euphemism of your civil liberties, right?
It's a stretch, but
it's a stretch, but that's the rationale. Is it's a euphemism, analogy, whatever you want to call it, simile, metaphor for your civil liberties.
It's unfortunate. I mean,
yeah. And
people don't learn from other countries, like Hong Kong.
People don't even read their own damn constitution, apparently. But yeah, no, there is this entrenched culture that started at the very beginning of the pandemic, where the recommendation was, gee, masks might be a good idea, and because that recommendation came from scientists and public health experts, it was summarily rejected by a segment of the population that had already been pre-programmed to reject anything that came from a view of expertise or maybe from the Northeast.
Yeah.
The public health people, instead of just coming out and apologizing and saying, I made a mistake and I should have said at the very beginning, wear a mask. They came out and said masks don't make any difference or they don't work.
There was mixed messaging at the beginning.
Right. And as my mom would say, really? Because they're working perfectly fine in the OR. And, so it doesn't make any sense. But when you come out and you sugarcoat it or give some kind of loop around and all this other crap, middle America doesn't have time to go through all of the bullshit of your apology. If you just come out and say, I'm sorry, I made a mistake, the argument ends. You don't have a rebuttal.
Right. But the masks are symbolic of a different problem. Yeah. Okay. So that might have helped, but the masks are symbolic of a much deeper problem, which is the knee-jerk reaction of a certain group of political followers to any kind of authoritative message. And by putting on the mask, you are, since it's visible, whereas standing 6 feet from somebody is not as visible, that became a visible marker of where you stood on the political spectrum. And that's where things really went wrong.
Yeah, but you wouldn't have gotten that far if you had just said, I'm sorry, I made a mistake.
Yes, you would have gotten that far. We still would have been in exactly the same situation.
I mean, our leaders, would still say, although they use that as an excuse. Yeah. Anyway, let's do two more.
One would have thought one might learn when they get infected and then need to go to the hospital because they're afraid that they can't breathe properly. And then
reality does not play into it. This is an alternate reality that people are choosing to live in.
Well, in that respect, one of the problems, in quotes, if you like, is that the virus is not actually lethal enough. There are too many people around. The vast majority of the population has no direct experience with it, and so it's hard to connect.
But there are now that we're approaching 250,000 Americans dead with this virus. There is a very, very large population of people who do have, if not direct experience with it, family or friend experience with it that is seared into their memory. And this, I think this virus has made a strong impression on a lot of folks.
Well, I think the number of 250,000 people, if you put that in perspective, that's more than Vietnam plus more than World War II. And you could say that's almost half of the total number that died in the Civil War, but not the percentage of the population.
Right.
So it depends on how you want to number crunch it, right? I mean, it's like we heard the government say, oh, we've done more testing than South Korea. We've done 9 million tests. Well, if South Korea has 30 million people, 9 million tests is a third of the population. If you're at 300 million people, 9 million tests is 3% of the population. Yes. See, we don't analyze the data using any sort of tool. We just yeah, I think we all need to take a course as a country in data analysis. Sign up, data analysis for dummies.
Not happening. Not happening.
I would thank Anonymous for the nice description of what went on in Hong Kong. It's a good lesson.
All right, let's go some picks. Let's do some picks, not go some picks.
Yes.
Amy, what do you have for us?
So I have a children's book that talks about the virus and actually though it's entitled Coronavirus, it talks about virus. It begins with viruses in general, about how not all of them are detrimental. Many of them are beneficial. And then it talks about you should wear a mask, you should wash your hands, why you should wash your hands, you don't want grandpa to get sick, blah, blah, blah. And
yeah, let's take care of grandpa.
Yes.
Good.
Grandpa or, somebody who's sick. Like my family is very conscious of washing their hands, wearing a mask around my mom who has starting lymphoma treatment. So, and I think it's a nice description with cutesy little pictures that takes out all of the politicalization. Like there's no description of the fish market. There's no inference of, it started in China, it's the Chinese virus or whatever. Right. So I think it's very nice because it takes out all the crap and it just gets to the heart of what you really want a kid to understand. Viruses can be good, can be bad. Wash your hands for this one.
I have a podcast. This is, it's called Lost Prologue, and the creator of it is Margot Gray, who is, I guess she just finished up at Duke University, where she majored in journalism, and she's done some stuff for public radio. She's a, an audio reporter. And she's done a really bang up professional job on this podcast, which is currently 3 episodes about the 1918 flu pandemic. And it is really, really good. And of course, longtime listeners to TWiV don't need me to tell them what the 1918 flu was about, but this was the pandemic. It was massively lethal to huge numbers of people. Unlike SARS-CoV-2, it preferentially killed the young rather than the old, and had an immense impact on world history, hastened the end of World War I, led to huge changes, which, and of course, most of the lessons we have since forgotten. But she interviews some genuine experts. She also interviewed me, so you'll have to sit through little bits of that. But she talks to people who actually know what they're talking about in terms of the 1918 pandemic, and it's just a really good podcast. So Margot's going to get a double TWiV bump here because you haven't listened to the last episode, but I picked this last time, Alan. Which is great. No, it's good. It's all good. Okay.
Give Margot a double TWiV bump. And I want to, I'll be repeating myself, but I have to tell you, Alan, because you'll appreciate this, that I was listening to this and this voice came on and it sounded really familiar. And I thought, but the interesting thing was my reaction was, that sounds like Tom Brokaw. And I thought, wait a minute, Tom Brokaw can't be here talking about the pandemic. And then it took me a few seconds to, it's Alan Dubb. So the message there is, you sound like Tom Brokaw. I never realized that before.
Yeah, it's good. Good to pick it twice. Rich, what do you have?
I have a vaccine tracker that was that's from a group called, the Regulatory Affairs Professional Society, which is the largest global organization of and for those involved with the regulation of healthcare-related products. This was I learned about this from my work with the Brighton Collaboration Vaccine Group. There are lots of vaccine trackers out there, but if you hit on this one and scroll down a little bit, there's a table that you can come to that is sortable by candidate, mechanism, sponsor, trial phase, or institution. The default is the things that are in the most advanced stages of trials, with links in each case. And what I really like is that if you click to expand on the left column, any one of these gives you a nice little summary of what the vaccine is and where they are in the trial.
Nice.
So I found this a very useful site. Very nice.
1, 2, 3, 4, 5, 6 in phase 3, 2 in phase 2/3. And I like this one here it's an inactivated vaccine from the Wuhan Institute of Biological Products. No name announced. How can you not name it? You got to name it something. Rich, by the way, the mic you're using, can you turn it so you're talking on the end more than the side?
Sure.
Because when you turn your head, you tend to get off axis,
okay.
you know? Know what I mean?
I know what you mean.
All right. I have a pick in honor of the Chemistry Nobels. It is A Crack in Creation by Doudna and Sam Sternberg. And this is a book that recounts the discovery of CRISPR and what Doudna did in her lab. And it's written by Sam Sternberg mostly, who was a PhD student in her lab, and he took a year off to write this. And it's really well done. It's nice history. And then he went on, he said he got tired of writing and he went back to the lab, and he's actually now a faculty member here at Columbia. The weird thing is it's written in her voice, but he wrote it. So that's weird. But I like the history. And then also TWiV 184, which is a podcast that I did with Sam shortly after he arrived here, all about CRISPR-Cas9 and gene editing and so forth. So we'll check that out. I think I'm going to re-release that on YouTube this weekend to get it more listens, because more people. It's a really good episode. Check it out. All right, that is TWiV 671. The show notes are at microbe.tv/TWiV. You can send us questions or comments to TWiV at microbe.tv. And if you'd like to support us, go over to microbe.tv/contribute. Our guest today.
Or Parasites Without Borders.
Yeah, if you want to have a tax deductible.
If you want to make a tax deductible contribution, you can go through Parasites Without Borders.
Parasiteswithoutborders.com. Our guest today, Amy Rosenfeld. She is ABR22 on the Twitter. You should follow her. She needs more followers.
Oh.
Thank you, Amy.
I think I'm following you.
Yes, I think so.
Yeah.
Thank you, Amy.
So. I'm very busy. I'm not that good at the Twitter.
Yeah, I know. You're in the lab all the time.
My plaque assay didn't work today.
What didn't work?
Sad face.
Your plaque assay didn't work?
My plaque assay didn't work today. Sad face. How many of those could you post?
That is one of the saddest things you can report. Because a plaque assay is the greatest assay ever developed, right?
Yeah.
Exactly.
I have to say, Amy has drunk the Kool-Aid with plaque assays. Absolutely.
Well, we have a collaborator who said to me, how many can you do? Because we're going to do epitope mapping for EV68. And I said to him, I do between 500 and 1,000 a week. And he said, is that automated? And I was like, no.
You mean plates?
Yeah, the box is 500 plates in the box. And I go through 2 boxes of individual 35 millimeter plates per week. That's 1,000 plates.
Well,
so we used to have this competition in my lab about how many plaque assays you do and how many you can do in a day and that kind of stuff. And it comes down to among other things, there's how do you define a plaque assay? Okay. And in other words, is it a well that you seed? Is it a plate that you seed? Or is it the dilution series? To me, a plaque assay is the dilution series. Yeah. How many dilution series have you done?
I agree.
Well, when you're a lab of one, the way it works is while you're incubating your diluted series of plates, you make new plates for tomorrow.
Oh, yeah. Sure.
So
listen.
I'm not clear.
Tech.
I'm not clear. Then I would be doing double the amount by this.
Tech at microbe.tv, right? Tech at microbe.tv. Get Amy some help so she could do more plaque
I only
acids.
count the ones, that are fully go into the incubator to incubate.
And if you get the job as a tech with Amy, you'll get to do lots of plaque assays, and you too will appreciate how awesome they are.
Thank you, Amy, for joining us.
My pleasure. Anytime.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com. He is Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. Siren.
What happens when you're at a medical center?
This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.